Hello, check. Hello, hello.
around the campus. Good afternoon, everyone. We already have our guests and the panelists here, but sorry, dignitaries, we'll just take 10 more minutes of yours. I know your time is very, very important, and equally for us, you are important. So please give us 10 more minutes. We have a couple of people joining us immediately. They're just around the corner in the campus. We're just dragging them in here. So sorry about that, but yeah, thank you for your patience. Good afternoon, everyone. So I know all of us are back after a lunch, or in fact, in after a lunch. So we will definitely make it sure that our panelists are going to grill up the discussion so that we get our food digested well. We have more people joining us in the due course, but nevertheless, let's start off today with this event. I am Dr. Srija Narayanan from the Cochin University of Science and Technology, and I have been given this privilege to introduce to you this event this afternoon. And this is an event which is sponsored or jointly being organized by the RGCB, the BRICS RGCB, and the Krebs BioNest. And this particular event is named the BioE3 Panel Discussion and Ideathon event. This is in alignment with the National Policy for Biotechnology. So today, we're gathered here to foster discussion on high-performance manufacturing and to inspire fresh, actionable ideas through our ideation event that will follow up after the panel discussion. We are honored to have with us some of the brightest minds in Indian biotech industry, innovation. So let us begin this exciting journey together to strengthen our policies and to strengthen our innovation temper in India. So the first segment would definitely be to inaugurate it. And for this, it gives me immense pleasure to invite our chief guest for the day, Shri C. Balagopal IAS, chairperson of KSIDC, a visionary leader who has continuously supported innovation and industrial transformation in Kerala. Most interestingly, Mr. Balagopal is the founder of Pen Paul. This is actually a blood bag manufacturing company that began very humbly back in Trivandrum and grew into the world's largest producer of blood bag systems, now exporting to more than 50 countries. That's a huge feat by Mr. Balagopal, sir. And then, along with his wife, Ms. Vinita, they have actually established and run a trust called Anaha. This trust supports socially beneficial programs in the fields of primary health, primary education, and rural livelihood. I call upon Sir Balagopal, sir, to please come forward and inaugurate this event. Please. Yes, sir. Thank you, dignitaries. May I now request Sri Balagopal to kindly inaugurate the session and share his insights with us.
Good afternoon. I thank Dr. Ramchand and others for having invited me to participate in this important event here, which is, as the panel discussion on advancing India's bioeconomy, focus on novel biologicals and biotherapeutics, and with an ideathon, which I think many of you will be participating on high performance biomanufacturing. So I just a couple of words here, which I have to say here is the, just like many things happening in other states and other countries, especially most emerging markets, biomanufacturing and biologicals is something which everybody wants now, you know. There is a story which comes to mind about before independence in the legislative assembly in Travancore, there was one particular very active legislative council member who was very active in seeing that whatever was decided, his community, because I mean, everybody represented one community in those days. So he wanted to ensure that his community also got its fair share. So he mostly didn't take much interest in the discussions and he'll be dozing off. But when it comes to an allocation or a decision, he'll put his hand up and say, we also want one. So one day he had dozed off, but the discussion was about a new cage for the animals in Trivandrum Zoo. And the decision was about to be taken when one mischievous member sitting next to this guy said, Ashane, very decision edukam povanu ningalkum venda idu. So he jumped up and said, I definitely demand that our community also should be represented and we should have one. So biologicals, biomanufacturing, semiconductors, everybody wants now. You know, it's the flavor of the month. Everybody wants it. And you will hear people in governments and every newspaper you pick up. You know, like somebody was telling me recently that when I saw a position paper which was sent to me in my capacity as chairman of KSITC, I saw a very scholarly note on semiconductors. And I was thinking, what are we in Kerala going to do in semiconductors? So I rang up somebody who is a world famous authority. I happen to know him. His name is Mr. Rajamanikam. And he is an internationally renowned expert on semiconductors. So I called him. And even people in Taiwan and all consult Mr. Rajamanikam. So I called him and said, what do you think, Mr. Rajamanikam? Do you think this has got any relevance for Kerala? So he explained to me about semiconductor manufacturing, that starting from the fabs, which require investments running into dozens of billions of US dollars, massive. And they require huge quantities of water, huge amounts of energy, etc. From that, you got a whole supply chain, which goes back to design. So he said you should select some place in this food chain, so to speak, and develop some competence in that. Don't try to do everything. Because he said today, in fact, he told me this. He said today if you go to Taiwan, at the Taipei International Airport, you will hear every Indian language spoken there. Because there are delegations from every state government which has gone there telling the Taiwanese, please come to our state. We are the best for semiconductors. We will give you whatever the others have offered you, we will give you 10% more. So right now India is passing through that phase. All very good, very nice, full of energy. But I only hope that at least a small part of all these resources being spent will be productively used and not just lead to delegations going and coming and nothing concrete coming out of it. So biologicals is I think has not got the kind of importance which it deserves, either in this country, this state, or in most of the states of the country. It doesn't seem as, what shall I say, as glamorous, as headline grabbing as semiconductors and these other things. But in my opinion, it's going to be probably far more impactful in terms of providing better livelihoods, much more remunerative jobs, much more value-adding, production of various very important products, etc., in the years to come. So Kerala, like all the other states, we've also drawn up a roadmap for this. And KSIDC is one of the agencies which is tasked with executing this plan. And as part of this plan, there is a life science park which is a subsidiary of KSIDC, which is coming up at Thonakal, a little outside Trivandrum, and where already several companies have started putting up their facilities. A few have already started functioning, and that is the same campus where the Institute of Advanced Virology is already working. So now the other thing which I want to say is that most of this initiative for focusing on semiconductors or whatever, these high profile technologies, people seem to think that it is a question of money. It depends on how much money you can mobilize, how many big name companies you can get, etc. But I think all of us miss one point is, how can you do all this unless you also import the skilled manpower, or you must be having the skilled manpower people here. So if you have to have the skilled people to run all these complex processes and facilities, etc., you need a higher education system which produces a strong pipeline of all these people. And that's where mostly the failures in India are. We still think of economic development and industrial development and high technology as investment driven. We think it's a question of billions and thousands of crores of investments, etc. But if you put up all these spanking new facilities, who are you going to get to work inside them? This will be almost like what happened in the United States of America, where thanks to Biden and his Make in America initiative, followed by the present US president and his Make America Great Again initiative. So they said, we are going to bring semiconductor manufacturing back to the US. And they spent billions in setting up this huge facility outside Phoenix, Arizona. And then the Taiwan Semiconductor Manufacturing Company found it doesn't have the people to run those factories. And they had to get the workers and technicians from Taiwan. Can you imagine? Who would have thought that the United States of America is going to have a shortage of skilled people? Yes, they may be having enough people for running the McDonald's and the various other things, but to run a highly specialized semiconductor manufacturing facility, they had to import people from Taiwan. And actually that's a disaster. That investment is a disaster for TSMC. So my point here is, we in India, in all the states, including Kerala, have to be very careful about these kind of plans. And I think the good news where Kerala is concerned is that for the last 10 years, some investment has been happening in the higher education sector, in building capacity, in investing capacity into creating research facilities in chosen higher education centers, both in the public university space as well as in private institutions. I have had the privilege of visiting some of them, and I know that some really cutting edge work is going on. And papers are coming out which are finding mention in the Nature Citation Index. And, in other words, they are peer reviewed stuff. So it is not just the quantitative things which are happening. So I think we are doing the right things. We have to also choose. Historically, if you look at it, Kerala has had a connection with biologics for a very long time, isn't it? Our herbal remedies and our natural, nature-based treatments and the great tradition of Ayurveda, which has found its finest flowering probably in Kerala, is all because of this very close relationship which we've had. Now by applying modern science to this, by marrying modern science, modern technology and modern clinical research to this huge empirical knowledge which is here already available about the local biological resources of the state and the biodiversity of the state, I think we are in for some very good times. And of course, we have great knowledge resources in the rest of the country. National Center for Biological Sciences is represented here, as well as other leading institutions. And it's a good thing that the private sector is also getting involved in Kerala. That's what gives me the greatest encouragement, is that the C.V. Jacob Center for, what is it called? I forget. Synthetic Biology and Biomanufacturing, which has come up here in the QSAT campus itself. And similarly, another one by Agape Diagnostics are setting up their own, they're setting up centers of excellence. So it's good that private industry, private business is beginning to find it worthwhile to set up such centers of excellence in public universities. So I think we are in for some very good time. I've taken much longer than what I expected to take for what I thought was going to be a very brief inaugural talk. So once again, let me wish this this seminar and this panel discussion and this hackathon all success. And I'm sure many interesting ideas will be thrown up, which will be of great benefit. And I hope Dr. Ramchand and the other organizers of this will send us a summary of the proceedings along with your recommendations for what the state government and KSIDC should do. Thank you and wish you all the best.
Thank you so much, sir. It has been a pleasure listening to your wisdom and advice. I'm sure the youngsters here, as well as people of our generation, are going to make use of your knowledge and your experience to gain our own ventures, to gain our own momentum. So here we have to now warmly welcome each one of our panelists here. Let me start off with Rajagopal sir himself. As I just mentioned to you, Balagopal sir. Yeah. Sorry, sir. Thank you for all that. So, welcome, sir, again. Yeah, please. Then we have with us Dr. Gopala K. Dasika. Basically, Dr. Dasika is a scientific strategy advisor for life sciences in the government of Tamil Nadu. And he's also the board member at the University of Wisconsin-Madison Global Health Institute. Welcome, sir. Next, we have with us Dr. Nitesh Dave. He's basically the former senior director of Biocon and is a freelance consultant operating in Bangalore currently. Welcome, sir. Next, we have Dr. Partha Hazra. He is the R&D Center of Excellence lead for Biotherapeutics at Biocon Biologics. Welcome, sir. Next, we have Dr. Radhakrishnan E.K. Sir is Associate Professor at Mahatma Gandhi University campus, and he's the Director of the Business Innovation and Incubator Center, as well as Industry Academia Interface Center at the university. Welcome, sir. And lastly, but never the least, my mentor, Dr. Valsama Joseph, Professor and Director of the National Center for Aquatic Health Animal Science. And she's also from Cochin University. Welcome, ma'am. But then we have the main man here, the moderator. He doesn't want a bouquet, but sir, would you like to take it over from me for the panel discussion? So that is Dr. Ramchand, sir. Thank you, sir.
Thank you. Good afternoon, everybody. Thank you, Dr. Balgobal. I know that I have called you in the last minute, day before yesterday, and so is all the people. So is Dr. Dasika, Dr. Partha, Dr. Nitesh, Dr. Radhakrishnan, and Dr. Valsamma. And thank you for organizing this and, inviting me to coordinate and do that. But you did a wonderful job. And the last minute, we also invited Srija to compare the program. So my job was to, to coordinate the panel discussion, monitor that, mentor that, or, so ask questions to the panelists. So before that, I was told also to introduce the subject. Basically, today's discussion is going to be on biotherapeutics and novel biologics. I'll come to that. And before that, what is the background also I need to explain to you. But we expected much more students here. But I understand that all the students have gone because of Onam, and tomorrow is holiday. And anyway, but then we have not given only one day, 24 hours for people to come. That's why, but at least some few people are there. And they all reached today, I think, from Bangalore. And they called them, immediately booked the flight and came. Because luckily I knew all of them very well. So that's why I decided to come. But we could not also talk after reaching and what is the questions, etc. So the questions will be very random. And before that, what I'm just wanting to take 5 minutes of yours to discuss the topic. You all heard about bioeconomy. I'm sure that what exactly is bioeconomy. And there are 3 buzzwords are there. BioE3, which represent that which economy and environment and education. That is what BioE3. And what has happened is about, I think, 2024 August only that government put up a rule regarding that BioE3, and they wanted to invest a lot of money for biomanufacturing and biofoundry. I don't know, several thousand crores has been earmarked for that. And what are the things government is going to be done, that also I will just briefly tell. And what can scientists do, what can entrepreneurs do, what can companies do, what can government do, and all those things we will discuss during our panel discussion. Now, what is bioeconomy? Bio means biotechnology, biotechnology-based economy, which actually, if you go back, really it will be connected to plant, it will be connected to microorganisms, and it will be connected to animals. These are the 3 major things, biotechnology-based, that, products or raw materials which we are using. For example, if you are talking about plants, which give food to us, isn't it? But it is not only that. Many, many other things can be done. And how a circular economy can be developed. Let's say these 3 things, plant, animals, and microorganisms. I will tell you a simple example so which you will understand. You might be knowing about chicken. And the price of chicken is probably about 200 rupees or something. I was just asking my driver what is the cost of chicken, and he told about, like, one chicken will be about 200 or 250 rupees, something like that, roughly about 100 rupees or something. which will give about 50, 60 or like per kg of the protein base, or the tissue or other things will cost about 70, 80 rupees. Okay. So I'm coming to the point. Do you know about maybe 30, 40 years also this was the price? How this has happened? Layer and broiler chicken now can grow in 36 days. 36 days they become 4 or 5 kg. And how this is happening? The food they are given is about 20 rupees per kg. And they are standing and going on eating and, putting on weight, and that is why the price is so low. The reason is that the world, a large amount of food is consumed in chicken, because every day 300 million, we are about 7.5 billion people are there, 300 million chickens are killed. And that is a very big industry. And where is the food coming from? To eat and make the appropriate protein, the food is coming from invariably wasted, plant food. So you can't take very high quality protein, high quality carbohydrate and vitamins, minerals, etc. So instead of that, very low quality, and it is formulated appropriately and given to the chicken. And the chicken, the fecal matter which is coming out also is utilized. Now how this is done is that, the various methods are there. 20 kg rupees per kg. Chicken feed is an amazing science. And there is something called FCR, feed conversion ratio, to make it to the maximum. That means if they eat 1 kg, at least 800 grams or 600 grams of the chicken, this, yeah. Okay. I'll come back here. Yeah, okay. So I was just telling about an example that, how that circular economy, bioeconomy can work. That means nothing is wasted, So the same way, and the same, we never knew what is to be done with the microorganism. Now microorganism, you all know about, the role of microorganism, gut microorganism and various microorganism, probiotic, how it is used in agri-foods and in human health, animal health, etc. So this is what exactly is called the circular economy, and without wasting anything, and this represents bioeconomy. And as I mentioned, 3 major things are economy, environment, and education. Now the other thing is that, biomanufacturing. So biomanufacturing, I will tell you a little later that why so much important biomanufacturing. Without that, many things can't be done. Now let us go into the impact on the Indian economy. You know that Indian economy is one of the economy which is probably growing at the highest GDP, gross domestic production, which is probably running around anywhere between 6 to 9%. Nowhere in the world it is happening. And our current the contribution from bioeconomy, somewhere in the range of 4 to 5%. It is very less. It can further be increased. And if you see about pharma, biopharma, it's only 0.7%. There are countries, like for example Switzerland, it is contributing 30% to the GDP. In US is about 2%. India is 0.7%. And we can really just increase that one. And bioeconomy in India is expected to go to 300 billion by 2030. How this can be done? That's why this whole exercise, this BioE3, we are behind and running around, and what is the role of biomanufacturing? As I mentioned, the most important thing what today is discussed is that biotherapeutics and the novel biologics. Now, the other thing is that, role in education and entrepreneurship. With all said and done, you know that we had only 50 biotech industries, 50 biotech startup about 10 years back. Now it is 10,000. That is happened in 10 years' time. Now, if you see about biotech means, I'm talking about the hardcore biotech. One is recombinant DNA, then all other vaccine related biopharma, then fab, various type of antibodies, vaccines, etc. These are the hardcore, and there are, what I understand is that about 10,000 similar such startups are there. Another disappointing thing is that out of this 10,000, normally if you see world over, about 2% or 3% will be successful. Let's assume that 10% is successful. There will be 1,000 industries will be there. If it is 1% successful, which is definitely going to happen, that means 100 industries will be successful out of this 10,000. Now, the problem is not that one. So we are talking about startups. Startup, everything is there everywhere. The small equipment, small facility, and you have scientists for that one. But that is not the problem, because I myself have faced that one. That's why I'm telling. This, after some time, after developing certain things, what you have to do is that, something called material generation for clinical trial. The first part is over. The material generation, there is only 3 or 4 places. Nitesh, I have discussed with you also some time back the problem. Only 4 such places are there which you can do material generation, and all these companies will require. But the most important thing is that biomanufacturing. Now, let's say recombinant DNA technology, if you are talking about the manufacturing, means what you require is that upstream, which is involved in all these, after the clone development, maintenance, and, fermentation process, etc., etc. You need the facility for that. Then another one is that, you have to purify the required protein in milligrams to gram to kg quantity from 10,000 to 100,000 liters. That is another, like the whole purification. Normal small acta and other things will not work, because all these things are there in all the industries. The same way, once that purification is done, it has to be formulated, and of course, manufactured, like, fill, finish, etc. is there. Where the people are going to be done. There is only one way. Either they will have to out-license, that is the practice everyone is doing, but then that is not the most successful way. If they have to do themselves, they have to set up, they need to have all these manufacturing, this facility, whether it is for piloting as well as manufacturing. That is why this bio manufacturing or bio foundry, which is being set up so that all the people will be able to utilize that one. Other one is that environmental sustainability. We'll have to see that, the environment is not killed. I will tell you how all these APIs, API means active pharmaceutical ingredients, or all the chemicals are synthesized by chemical methods. By chemical method, for example, 1 kg of an API is made, normally about 100 liters of solvents are used. So that is very bad for the environment. Now slowly this is changing into enzymatic method. That means you can do the whole thing in aqueous medium, and the whole conversion can be done. Everyone is trying to do that one, and for this various different enzymes. That's why the synthetic biology has come up. Exactly what is that, Dr. Nikesh is sitting there. So that is what exactly they are doing. The using synthetic biology, various enzymes are made and purified that can be used for various purposes, so that the environment will be protected. And eventually what will happen is that we'll be able to see a carbon negative. Right now it is because of tremendous oxidation of fuels and other things, a lot of carbon dioxide is made, but eventually we'll be able to see carbon zero or carbon neutral or carbon negative thing. So, which briefly I mentioned about biomanufacturing in India. And finally, the novel biologics and biotherapeutics current landscape, that is what we are going to discuss. But why this particular area is taken? Let me tell you, 2 more minutes I will take. Why RGCB has chosen that, I think, for the discussion. There is a reason. If you see, the drug discovery started probably about, the rational drug discovery started about 60 years back. Something or the other started, like, aspirin and quinine, those type of molecules started coming about 100, 120. It was not rational drug discovery, it was serendipity, and for 40, 50 years, slowly start understanding the mechanism. And last 30, 40 years, all these small molecules start coming. which I was mentioning about API. For example, your paracetamol, metformin, those type of molecules. And at the peak of the time, maybe 40 molecules came. In the last few years, only 4 or 5. Now that is coming down. Everything is biologics. Now, the reason I am telling that, India has to get into this, because we have not produced a single small molecule NCE till date. The time is over. Now it is the time for biologics. How these recombinant proteins can be used for therapeutic purpose. That's why government has decided we should not lose this opportunity. We should start making, because now I will tell you, next 20 years from now, these all small molecules will significantly go down, and it is going to be biologics. Right now, 80 different, whether it is antibodies or protein molecules, are in clinical trials. The rough number all over the world. But we still, in India, has to develop a novel biologics. That's why some of our company, especially Arunabha, is sitting. One of the company what I headed, called Saxin and Therajan Biologics, we developed 2 novel biologics for anti-VEGF for macular degeneration, wet AMD, and diabetic retinopathy. And right now, what we are setting up another facility over here, which is also related to how these can be targeted, and also how novel molecules based on the mutation, how this can be developed. So these are all comes under novel biologics and biotherapeutics, and I'm not going to discuss more about that one. And after the panel discussion, students and you can also ask questions, or in between if you want to ask any questions to the panelists, that also can be done. And I think we can start the panel discussion now. You already got an introduction of Dr. Balagopal. Dr. Balagopal, I think he will come back so we can ask certain questions related to, especially he has a good experience in developing a company which is called Penpal. As early as 1983, he was a senior IAS officer, which hardly anybody leaves an IAS position. At a young age, probably was 30, 35, and decided to start a company which became a very successful company. And at one point, they're sending this blood bags to about 50 plus countries. And he's chairman of KSIDC. I'm one of the director of Kerala Life Sciences Park, so I work with him. And the developing, that's why I am here connected to this. Otherwise, I'm actually connected with a few other companies. Dr. Gopalakrishna Desika, I'm not going to read out, but very eminent biotechnologist, biologist, drug discovery scientist. He was the senior VP for Pfizer. He was also chief scientific officer for Biotherapeutics, and worldwide head of research and development for Sandoz, plus several publications, patents, and very eminent scientist, and he is also an advisor to Tamil Nadu government in the biotech area, which I am also there, but I am connected to Anna University. So you can ask questions on drug discovery, development, biologics, vaccines, any of those things, and I have also certain questions. Dr. Nitesh Dave, I know Dr. Nitesh Dave from 2000. Or 1999 or 2000, I joined as Vice President of Sun Pharmaceuticals, and one of the earliest person I hired from Torrent was Dr. Nitesh Dave. But interestingly, I was always telling, at that time we did not do PhD, I was telling, you should go and do PhD sometime, isn't it? And which he did, and he became senior director of Biocon, but now he has his own company. And one thing I have noticed is that normally I will be very careful interviewing people, and one of the best interview I have done was Nitesh, because so much very in-depth questions I asked. Then I also told this to your at that time boss, Dr. your understanding. He also told the same. Thanks. So I know that you will be able to answer any of the questions from me or others. Dr. Partha Hazra is the also a pioneering bioprocess scientist and leading the Center of Excellence in R&D for Biotherapeutics at Bioeconomy. And thanks for this short notice and desire to come, and thank you in advance for going to enlighten us. And Dr. Radhakrishnan is very well known in the academic field. I know him for quite some time. And Dr. Radhakrishnan is an associate professor as well as involved in many things over there, especially startups. Then he's responsible for making, I'm a professor of practice at MG University, but I still have to visit there. That's the thing. Last 2 years or something, and I hope sometime I will be able to come. So I'm involved in like PhD college and all, but regularly I'm going there. He is going to discuss more of academic settings, setting up startups over there and related things. And in case the questions are not right, you are always welcome to discuss what you feel like. And Dr. Valsamma Joseph, and we also have talked couple of times, if you remember.
Yes.
Yeah. Because you know my cousin.
Generation.
Yeah. Professor Valsamma Joseph is a professor and director at the National Center for Aquatic Animal Health at Cochin University of Science and Technology, with a PhD in environmental science, and her research spans marine algal biotechnology, aquatic animal health, algal genomics, and metagenomics of biochemical cycles. So with this, we can start the panel discussion. So let's start with some general bioeconomy, and probably any one of you can answer that one. That's not is a technical question, but still, yeah. So what does the term bioeconomy means, and why is it relevant to today's world? So we would like to hear probably from you why it is important in today's world.
Yeah, first of all, am I audible? Yes. Yeah, thanks. Yeah, first of all, thanks Dr. Ramchand for the invitation, and really happy to be here with you all today. So why is it relevant today is the question. So I think you've spoken about bioeconomy and what impact it has. And in today, I think globally the economy is moving from more of fossil fuel based economy to bioeconomy, and it's important also because I think if anybody saw today's newspaper, Economic Times, I was reading this morning, Health Economics Time. In India, we are actually losing about 1,000 days of our life just by living in India, was the statement that was the title for the article. And I think it's got to do with the fact that we are not taking care of the environment. We are using fossil fuels. We are creating so much of pollution, and it is having an impact. So the whole thrust on bioeconomy is, I think China did this many years ago, and over the last 10, 15 years, they have grown to be the largest bioeconomy in the world. Okay, it's not the US. China is way ahead. And today we are, while we are growing, but we are long ways behind the rest of the world in this space. So I think it's important that we focus on using living cells, systems, processes, and develop environmentally friendly products, services to enable us to have healthcare solutions and improve our lives on a daily basis in a sensitive manner. And that is really what bioeconomy is about. And because of this general awareness, the climate change that is happening, and the government's policies that have been put in place across the world to reduce climatic impact, bioeconomy has become very important. And since we are so far behind, our government has taken an initiative to put in place this BioE3 just about a year ago, 24th of August, it was put in place. And I think that is going to help us guide our growth in a way that is going to be environmentally sustainable and put us on the right path for success. So I think today it is very, why is it relevant was the question. It's very relevant because we have been we have been doing things all along in a way that is so detrimental to the environment that by focusing on bioeconomy, we will turn the clock backwards and hopefully have longer, sustainable solutions for the world. So those are my.
Thank you very much. Yeah.
Just to extend to that, and I thought maybe it will be more relevant in addition to what Gopal has told. We all know that nowadays there's a lot of talk about the oil, which is a fossil fuel, and a direct impact on the environment of the fossil fuel we are all aware of. One of the classic example of that is if you have read the papers about blending of ethanol. And we all know the classical fermentation. Ethanol is the byproduct of classical fermentation, which happens through molasses. We were at about 2% blending at some point in time. And we have come a long way. Today we are blending at 20%. The carbon footprint of ethanol is way lower as compared to what environmental impact something like gas or petrol has. So that's something which is we are feeding fermenters earlier with molasses. Slowly we have started putting corn syrups and everything into it. There are initiatives I know aware of where companies been working on having other carbon resources to feed to the bugs to produce more ethanol. So there's a huge lowering of the environmental impact with the fossil fuel. They've also extended this particular blending to turbine oil in the aeroplanes. And I think we were at about now at about 0.2% mixing. They have an ambitious project to make it to 2% in the next 5 years. These are direct impact of having and replacing something like fossil fuel to something which is an alternate energy source.
Yeah, probably if some time permits, I have a few other questions related to what we are telling, but then let's go to our other questions. And to you only, I'm just going to have 2 questions together I'm going to give, which is connected to biologics, biotherapeutics. So for the purpose of students, you explain to them what is biologics and biotherapeutics, that is number 1. Then for the development of purpose, developmental, how complex or how simple is biotherapeutics? That means biologics, novel biologics, or antibodies, or whatever it is, compared to NCE, new chemical entities. So what are the challenges? How you are able to overcome? So these are the 2 questions combined together. We can.
So I will keep it simple. Looking at the audience, what we have a lot of young people here. We use these terms very interchangeably at times. Biologics or biological drugs or biological products are nothing but how we utilize a biological system. It could be any biological system. Dr. Ramchand explained utilizing any biological systems like microbes. It could be animals, it could be tissue extract, it could be explants, it could be plant tissue culture, utilizing plants to do this. If you utilize any biological system for benefit of mankind, having any kind of product, or the services out of it, it is a biological product. When you talk about biotherapeutics, it becomes more specific. You look at microorganisms in the current context, and therapeutics means you use or make some of the drugs or the products for treating some of the diseases. And I will take the simplest example way back of a simple molecule called insulin, which was made long time back using porcine sources, or before that bovine sources. And they were simple extracts, and they had huge safety issues where the associated viruses or something will come along with it. But they were of biological origin. Nevertheless, with the advent of recombinant DNA technology, they made and cloned these genes early on in E. coli, and then they made a final recombinant insulin product by a company called Lilly. And this is the first, one of the first recombinant protein or biotherapeutics which was made. And we have come long way after that. Now we have products which are in various disease segments. Diabetes is one of the biggest one. We keep hearing about nowadays about the classic, the talk of the town about the Wegovy or the semaglutides or terziperetide, which are the new inventions in the obesity world. And you have a range of monoclonal antibodies which are being produced through mammalian cell culture. And there are novel molecules. They have gone a step further to make conjugates. We have ADCs, drugs now into the market. You combine antibodies either with the proteins. There are drugs which trying with DNA. These are all utilizing a biological system. You utilize other microbes, you utilize bacteria or yeast systems or mammalian cells. These are all the category of biotherapeutics. Now coming to the second question, how complex they are. We all know biological systems are complex. The processes of making these products, they are as non-linear as people like us human beings. We behave differently at every moment. That's how the bugs are also. That's a miniaturized system of a large multicellular organism. No single batch is identical to the batches which you have taken earlier. Role of a scientist or a biological or a biotechnologist is how to minimize these variations which happens in the biological systems, and to make a product which has a good quality, which has a good safety profile, and it passes the regulatory muster, so that and it is efficacious. Yes, there are 3 or 4 criteria for an effective drug. Now that is where the complexity lies. You touched upon a very critical aspect of the environmental impact. Most of the systems are making of the drugs in a biotherapeutic area versus chemical synthesis is mostly aqueous based. Even though there are solvent which is being used, there are technologies where you can recycle these solvents and minimize the use of these, while you use hazardous chemicals in terms of when you use a chemically synthesized product. Also in terms of development, if you have a abbreviated version of making these products, then you are just okay with either phase 1 or phase 3, or maybe sometimes you can get a waiver for a phase 3 studies. Whereas if you, and many a times the safety profile, if you have a novel biologics, you have to establish it, or if you have a known molecule, then you have already background information about how these products have behaved in different animal models or in vitro assays, or even at times in the first-in-human studies. So the pathway for making a biologics is tedious. Nevertheless, the safety profile of these drugs are super safe. If you take a monoclonal antibody, you can take for example trastuzumab, for example, for breast cancer. You can give the patient in gram quantities without having any adverse kind of impact. And as compared to if you take any other chemotherapy drugs, for example, we know what kind of side reactions you have it. Safety is a big, and the tolerance of these drugs, the biological drugs, is much safer as compared to chemical drugs. Regulations in terms of knowing how the biologics behave, it has come a long way, and mostly led by European Union. They had this policies of approval of these products way back in 2004, 2005. They made a very clear-cut guidelines about how to make these products. And now we have come up to a way where you can get product approvals in Europe by doing a simple phase 1 studies. We are just getting into like a small molecule, just doing a bioequivalence kind of a studies. We are almost reaching to that stage. Some countries have done away with preclinical studies, so no use of animals, for example. Recently, FDA came up with a guideline to have a waiver. So there is a lot happening. There is a long way to go. But as you said, quite a few products today are being approved, which is of the biological routes, because of these reasons which I explained, for a few minutes.
So touching on your questions, let me also tell you that, as he mentioned, there are two different types, NCE, new chemical entity, or novel molecules. It's called small molecules. The other one is the large molecules, protein, recombinant proteins. And what I have done, I was involved in doing small molecules when I was in Sun Pharma, but it is far more complex, let me tell you, because thousands of molecules you have to synthesize to get the right pharmacophore. And the screening procedure is very complex. And eventually, let's assume that you are getting a molecule, finally go into clinical evaluation in phase 1, only 20% get out. That means 80% after getting into the clinical trials. The complexity is so high, that's why till date we could produce only 450 plus molecules. That is absolutely novel energy. There is something called Me Too drugs. There is 2,000 plus is there. That's about small molecule. As far as large molecules, my experience is that, of course, the method of developing is start with something called phage display technology, which will give all different types of configurations and conformations of the protein and, screen and develop. But one good thing, as he mentioned that, because many of these biologics is very safe, many times, because it is biologics molecule, protein molecule, unless it produces antibody against that one, is invariably fine. But the difficulty is that early part, the cloning and expression and the purification part. Many people many times fail that. At least small, I'm not telling about when you are doing biosimilars or something. This is what we have seen. But when you have passed through that, if you are getting into clinical trial, the chances of failure is extremely small. So that is one thing I just wanted to.
Can I maybe?
Yeah, please.
If there is time, I don't know. Yeah, just
We have sufficient time.
Yeah.
not
Okay.
worry.
Yeah, I just want to add to what both eminent speakers have said. I think just to simplify this, we can think of drug development or biologic development, small or large, having two parts. One is discovery, the other is development, right? And so if you look at the timeline that is needed, it would be anywhere upwards of 8 to 10 years, maybe 12 years for the whole process. And when it comes to small molecules, as Dr. Ramchand mentioned, the discovery part is pretty complex because you have to screen a large number of molecules, and then you have very, very small numbers where you get potential leads that you want to pursue, and then many of them actually don't make it. And it's not so different even in biologics in terms of the discovery part. But I think that the complexity with biologics is in that there is a whole range of products that fall under biologics. So I think what has been illustrated in the earlier conversation largely is about proteins and antibodies. But you also have other forms, nucleic acids and complex molecules, which are like proteins linked to a toxin, ADCs, or even cell therapy or gene therapy. So when you look at this whole range of biologics that are being developed, the complexity is many fold more. I think that is where the challenge is, to ensure that we have appropriate processes, appropriate regulation, appropriate methods for each of these complex platforms of drugs that we are looking at now, and ensure that efficacy and safety is met in all these cases. And I think that is where I think there is orders of magnitude higher complexity and cost and technically challenging areas that you see in the biologic space compared to small molecules. The other thing is about developability, right? So even with protein therapeutics, the stability is a challenge, and access and so on. So developability of a larger molecule tends to be somewhat more challenging. So these are the two things I wanted to add.
Yeah, next question is to Dr. Partha. Dr. Partha is from...
Before we go to the next question, I'm just add one more point is that, see, we keep on distinguishing between the biological products and the chemically derived product. But irony is that today there are drugs which are getting developed, which is called the antibody drug conjugate. You might have heard about that one. This is an ideal example where the biological molecule is merged with the chemical molecule, and finally both in combination the final product is actually the therapy. The biological molecule means antibody gets merged with the potential chemical molecules, which goes as a chemotherapy as an example. And antibody we know for the directed therapy, right? And chemotherapy is indirected but effective therapy. So combination of those two is a directed effective therapy, which is called an antibody drug conjugate. So we cannot distinguish now anymore about the chemical molecules and the biological molecules. Just as an example I am giving. Such type of things are there. Earlier also it was there. It is not that antibody drug conjugate is the first one. There are multiple other molecules. Just as an example is the, I would say, PEG filgrastim. PEG GCSF it is called. GCSF is the biological molecule. But when it's attached to the PEG molecule, to increase the half-life of the molecule inside in the circulation, so it is called the PEG filgrastim or PEG GCSF, which is much more effective than the normal GCSF.
Yeah, in antibody drug conjugate, one of the other challenge is that, when you are linking the small molecule, because the small molecule has to go inside the cell, cancer cell. The thing is that, antibody goes and binds to that one. And you need to identify the right enzyme which breaks. Now, the whole thing goes inside, it will not be active. Now, if it breaks, the conformation, that means if simple change in one single covalent bond changes, the conformation will change, the drug is not effective. This is one of the things many people are actually facing. I was wondering, there are other, because I'm working for the same way, reversible binding, can be done. You're just going to works here, the same one, instead of, antibody in a different way, which we will discuss later. So now my next question is to you.
Just one more point, I'm sorry. So the breaking is extremely important part, whatever you were saying. See, for the antibody drug conjugate, if you read or if you are interested, you can see that there is a linker is also attached with the drug molecule and which is coupled with the antibody. So that helps in those type of things for the breaking of antibody and drug molecule, or maybe going into the recirculation and whatever it might be.
There are several strategies.
Strategies
Yeah,
are
I know,
made,
many, yeah.
There are an enzyme cleavable thing and non-enzyme cleavable multiple.
You can have the PEG, which is affinity for intrinsic protein. Your classic example is drugs like Wegovy or semaglutide or maybe terziperidine. They have a propensity to go and bind to albumin in the body, and it's a covalent bond. So an albumin is an abundant protein, so it extends the half-life to an extent that you can. Some of these formulations are also you can take orally. So that's an advancement in these kind of linking technologies.
So now let's go into, now I will come to maybe, so the other area, probably fisheries, agricultural, and other than biotherapeutics. So if you have any points to tell yourself, that's fine. So otherwise I will ask questions. Yeah.
Yeah. So first of all, I would like to express sincere thanks to Dr. Ramchand and also to Dr. Divya Purushottam for inviting to this particular program. So I would like to share some like points, especially to the beginners. And what I was thinking is about what is the basic philosophy behind BioE3? So that's the basic question I was thinking, especially that is very important, I think, for the beginners. And what I think is that the entire responsibility of the nature is knowingly or unknowingly entrusted to human. So we have to take care of the entire nature in various ways. So from that point of view, whatever thing there that we can experience as problem or demand, and all these things are to be solved. So the problem is of biological origin in some way, or in most of the ways, or it can be linked or aligned to the biological origin. So from where we can find the solution, that again we have to find from biological origin. So a problem of biological origin, where a solution should be identified from the biological origin. And by connecting these things is a very basic logic, I think. And in that point of view, from the beginners, especially from the student community, or who are just into this area, from any branch of biology or any subject, and these simple realization is important, and also the changes are to be introduced in our academic and other learning and teaching activities in various ways, that all these process should some way get aligned in such a way that there should be a learning process which is aligned with this requirement. And when the learning process is aligned with this requirement of BioE3, then it will become much more productive and as expected by the country. And there are several changes in our learning pattern itself is important, because from theoretical learning to real world problem, that is very important. Exposure to various types of like experts, as we can see here, and like that we have to learn from experts in various sectors, and how we can learn from mentors. And also at the same time, the students should get exposure to how to approach a problem. So whenever a problem is coming to us, it is not like the way or the mindset we have to approach from the conventional pattern, where we are approaching a problem to write an answer, and when we think that that answer is completed, means our responsibility is over. So when we are changing into BioE3, we have to change our mindset from just writing an answer to generating a solution. I think that is very, very important. And also, there are many government schemes are there, and the students should get proper training to apply to each and every schemes. And where we need to arrange several training programs, then only the student will become competitive and capable enough to get into the main or the major flavor of the BioE3 policy. Because there are several schemes are there, and by simply applying and by getting some rejection, they will get disappointed. The thing is that they need to be provided with how to prepare a project, or how to present a problem, how to apply for a scheme, how to generate, and these are very important. And another important question is, now the question is, what are the areas that are coming under BioE3? If the question is like that, we can say that all the areas will come under BioE3. Because bio means we are doing, humans are doing, so it addresses everything related to human, or related to like environment, or related to nature. So it will come all areas, or any area can be. So who can make use of this one? Again, anyone who is interested in this can exploit this opportunity. So we need to so-called democratize the objectives and the ways how the BioE3 policy can be made in such a way that just like everyone is making use of smartphone, can we make the BioE3 policy in such a way that our students should be smart enough to explore the potential of BioE3. And I think that some action point in that way is very important, because so if we consider just like any production line producing any material or whatever it is, there are different stages are there. Finally, we are getting the final product. So the students are just like in that production line, they are in the institution, or where the college or university, wherever they are in the first stage of the production line. So how effectively and perfectly we can nurture them by giving all this flavor, that will ultimately matter the outcome of this policy. And this is a general like a suggestion I would like to share.
Okay.
Thank you very
So
much.
now I'll come to you, Dr. Well, Samma. So, bioeconomy also is connected to health of people, and
Yeah.
how the traditional method is synthetic method for drugs or pharmaceuticals. Now, largely also depend on other area. So, how you think for developing molecules or developing drugs or other things, how we can depend on agriculture, how we can depend on food and fisheries, especially the
Yeah.
fisheries,
Yeah.
marine,
Yeah.
marine
Okay.
products.
Okay, thank you. Thank you very much for this opportunity and for that question. Yeah, I will talk from in a different angle because we are a part of web of life where most of our technological innovations are focusing on the health of the human. And pharmaceutical industry is mainly dominating this. And we have other areas also. Especially the spirit of this BioE3 policy was government of India has been spending crores of rupees over the years on basic research in various aspects of life sciences. But even today, the biotechnology industry sector was not able to boost that much economy as well as generate employment for our generation, because we have been looking into the traditional job sectors like having a job either in an academic institutions or postdoctoral position, but why not many people have turned into entrepreneurs? This is a very important question. And we have been conducting this DBT sponsored MTech Marine Biotechnology program, the idea of which was to generate job makers than job seekers. But we have hardly, over the past 18 years, we have hardly 1 or 2 or 3 startups which have come up. Whenever we are motivating students, they are not able to take up that challenge of developing a technology or not. So if you look back at the human health sector, that is the ideal case for development of a technology. How? Because in any pharma development, we are looking at the patient. The discovery originates from the problems of that patient. We are understanding that problem, and then we are arriving at a final solution. A technology is derived and it is translated into the market, and industry is there. And but there are several other sectors also there where we are developing a startup and then we are imposing on a market or system. It will not be viable. There should be that sir was telling about that first discovery stage. That is very important. We have to understand the system in which we are going to solve. It can be veterinary medicine, it can be human health, it can be fish or shellfish, it can be anything, it can be plant health. But what is the requirement there? That system we should understand. Then we should develop the technology. Then comes who requires this? Because if we are trying to translate a technology to a farmer, whether it is in agriculture or in aquaculture, they won't be able to take up highly costly and sophisticated technologies. For example, in aquaculture, it's a very important issue that, overnight a massive fish kill or a shrimp, death occurs. And where is the solution? So there can be highly sensitive and affordable diagnostic systems made available, which will be, affordable for farmers, So it is into the sector. Even for human health, that is a very big issue where we have to look at the affordability of the larger population. So many of our students here, many of them are young startups, and for all that, you have to think in a different line, than what is that understanding that problem at base level, finding solution, then translating it into the, who is at the receiving end. So this study is very important for developing, a startup or any industry ecosystem, so that then only it will be able to bloom economy as well as, the employment generation. That is one thing. Another is coming to the environment. As we know, we are part of web of life. We are not in isolation. So there are several critical issues which we are looking at. One is
increase in carbon dioxide, and we are dealing with the marine biotechnology. When we look at the ocean planet, what is the most critical is the change in pH because of the increase in carbon dioxide concentration. So the life out there from biogeochemistry to any food web interactions, biodiversity, everything is changed. Another is increasing oxygen depletion in oceanic zones. And this is also leading to several changes. And this we have done loss of biodiversity. This is also very important. And so we have to develop technologies which is in a circular economy mode where there should be zero waste discharge. And right now we are in the process of developing some Indo-European consortium project on marine pollution, and also we can see that everything, any pollution which is there reaching the marine environment or any aquatic system, it can be terrestrial or brackish water, everything is coming back to the humans in a different form and leading to health problems. And again, we have to go for new and new innovations and interventions. So this interconnectedness, every, entrepreneur or every, aspirant to be an entrepreneur should keep in mind this interconnectivity. So then only we will be able to take care of this environment. So Government of India has identified bio-based chemicals and enzymes, functional food and smart proteins, precision biotherapeutic, climate resilient agriculture, carbon capture and its utilization, futuristic marine and space research for developing different translation level products, that is different maybe basic research, network research, filling the gap area. If you already have reached some advances, then you can do the filling the gap, and also having large biomanufacturing facilities in India and also biofoundry setups in India, where any startups can go and do their production process. So it's a huge mission. And we all have to work towards that in our own areas. So that is what I have to speak as an introduction. So maybe related to later,
we have,
specifically I can go.
Yeah, we have sufficient time. That's what I was asking the coordinators, and there is no problem. We have up to 5 o'clock and only few speakers for Ideathon, so we have sufficient time. Now, what I suggest is that, after one more question, I will come to students and the startup people to ask some questions. One thing what I have found is that wherever I go and give talks, not in Delhi, not in Bombay, not in Gujarat, but in South, especially in Kerala, no one asks questions. Nobody. It's like, I have my own experience. I did organic chemistry from Kerala. I never knew to speak English also when I did. No, I'm not joking. When I went to Baroda, only thing I got an extraordinary mentor. Professor Venki Ramakrishnan, Nobel laureate's father, he was even better than him. So somewhere or other he took me, even if I could not speak English. But the advantage there is that, make sure every Saturday there is a seminar, and the 2 weeks later, I was asked to give a seminar, and I was asked to teach students. They are brilliant students there. These students which I know nothing, microbiology. I was an organic chemist, never studied biology. Anyway, so what I'm telling is that the training, mentorship was extraordinary there, and students get an opportunity and they have to ask questions, which I have been asking many, many places. And recently from Trivandrum, I was called that, please, sir, make them to ask questions, what we should do. So you're going to come, please think about some questions, and anybody, all questions are right questions and good questions. It's not bad questions. So that's what some people are telling. They're worried. The questions is relevant. Doesn't matter. Every question will be having an answer to. So I'm just wanting to ask you, because Bioeconomy is the specialized, of course, novel biologics as well as biosimilars. So, you know by now what is biosimilars. It's like generic. Somebody also developed, patented, and the same thing, when the patent is over, which is developed in India, countries like India or other places, it is called biosimilar, similar molecule. And it has got its own complexity, because biosimilar development is not as simple as generic molecule, because generic API molecule synthesizing is not that complex. To develop that takes only few 6 months or 8 months, but biosimilars are different. Now, my question is very specific. You know, what are the challenges, especially, glycosylation part? Unless you use everything similar. For example, your medium and, same organism, same genetic makeup, the glycosylation can change, and you will have to show also where is the glycosylation points. Now, have you met with a kind of that issue where probably the activity will be same, the efficacy will be same, no toxicity, but your point of glycosylation is somewhere else, or different. So how you are going to convince the regulators, or does this has to go as a novel biology? I hope you people understood. There is probably you will explain that.
Okay, thanks. It's a very, I would say, very thoughtful question. See, first of all, before I answer this one, I just trying to say, what is biosimilar? Let us understand that first. Correct? I will take few minutes if you don't mind. So, what is biosimilar? In a chemical molecule, or maybe the chemical drugs, we are very familiar with the term that generic molecule. Correct? Rather, I would say very simplistic way that biosimilar is the generic molecule of the biological derived drug. Biologically recombinant derived drug is a biosimilar. Now, of course, a biological drug development itself is quite complex. I would say, and in the biological drug development, there are two parts. One is the novel biologicals, another one is the biosimilar biologicals. In the novel biologicals, you know that, yes, the molecule has to establish its, first, the function relationship with the body. That means for what purpose the molecule is being developed, you have to establish that. And then it comes as the manufacturing and all phase 1, phase 2, phase 3, phase 4 clinical trials. For that is the reason it is a very lengthy pathway, right? And it takes anywhere between 7 to maybe 15 years also for the novel biologics. Now, what is biosimilar biologics then? Biosimilar biologics, as I told, the product is already established. That means the product safety and efficacy is already established. You have to make the product which are similar safety efficacy profile. Safety and efficacy profile. Now, how you will prove that similar? Because now I am coming to the complexity of developing a biosimilar product. So, for the biosimilar product, you will get a benefit of having limited clinical trial, because you don't have to do phase 2, multiple phase 2 clinical trial, because it is established. So the main focus goes into the chemistry manufacturing, or the quality of the product. How good is the quality? Or maybe how similar is the quality of the product to the originator product, or maybe to the reference drug product, whatever we tell. Now, reference drug has been made probably with a certain process which has worked for them, and they have kept that process, because in the whoever is developing the novel product, the initial part of the product development, the process of manufacturing part of the process product development takes very, I would say, less importance. Rather, it goes that how to establish the clinical efficacy of the product. Right? Okay. So the product has been made for the novel biologics. Now you have to mimic the product quality, whatever maybe thoughtfully or without any thinking process the product has been made by the novel manufacturer. But mind it, as a biosimilar developer, you have to meet that product quality. Product quality means there are multiple purity, impurity, there are case of glycosylation. glycosylation has an individual role to play for the monoclonal antibody. At the same time, for the non-monoclonal antibody, glycosylation is immunogenic. That means it is not good to have. So you have to match the glycosylation profile of the monoclonal antibody, because each glycosylation has a different role to play for its function. Now the question comes, how difficult it is? It is difficult. Rather, the making, the lab making, or maybe the manufacturing making days in and out to meet the drug product which has a specific range of the specification, it is difficult. And you have to meet them exactly, irrespective of which organism you are producing. I give you one simple, very simple example, but very striking example. One particular product is developed by certain company in the E. coli. Correct? In E. coli, there is no glycosylation. It's a prokaryotic system. There is no glycosylation. Glycosylation is immunogenic. So, if you have a glycosylation in your product, it will be challenging. You will be questioned by the regulator because it is immunogenic. Your product has glycosylation, but the reference product or the originator product do not have the glycosylation. So what you will do? So in the process, suppose either you have to change the reference product, suppose you make the, you have to change the organism, source organism where you are making. If it is not E. coli, that is, if it is a say fungal system, yeast system, there is bound to be glycosylation because those are the eukaryotic system. So you have to take care of those glycosylation in your process of manufacturing. That means those glycosylation you have to remove to meet the quality. Now the question is coming, even if there is a certain differences, in the monoclonal antibody glycosylation is bound to happen and it has a multiple glycosylation, there's fucosylation, galactosylation, sialic acid, different type of glycosylation happens. Now the question is that do you have to meet each of them to be biosimilar to get approval? It is difficult, definitely difficult. It is good to have it, but it is not mandatory also. If it doesn't meet the exact glycosylation, because you are giving the glycosylation example, that's why I am giving the glycosylation example. If it doesn't meet, then you have to justify some orthogonal treatment or orthogonal testing that this glycosylation, I am not matching glycosylation A versus glycosylation A of reference product. I am not able to match exactly. But I am showing some orthogonal test that these glycosylation differences doesn't impact the safety efficacy of the product in human. Definitely, the regulator will accept it, provided your justification and scientific rationale is correct. So those type of challenges the biosimilar developer has to face. That means, as I told, the innovator may be making the product in certain way and that becomes the norms or that becomes their process. But irrespective of that, you have to make the process in whatever way you might be, but it has to match the quality. And also at the same time, it has to overcome all the patent, whatever you have it. We know the product. We have to do the characterization of the product in so detail. Sometimes we can claim easily, we know about the innovator product much more than the innovator knows. Because we have done so much of intense characterization, or the actual developer has to do that, to know that what is the quality of the product, what innovator has, so that you can meet the criteria.
Okay, I have a small question to Dr. Balagopal. I was telling about how, almost 45 years back, how one of the earliest, I would consider, though it is blood bag, it will consider as a biotech company. Definitely it will come under biotech company because, blood clotting is involved. You were a senior IAS officer and no one, junior, okay. but you were an IAS officer. And how challenging was that, especially that, because what we are finding is that, when you are entering to start something, is not the technology, but so many other things comes up and, so difficult. So what are the challenges? And, and of course, I know that this company became a very big company. Even now it is a big company and the world's probably one of the largest company, 50 countries you are exporting the product. But how did you do? What are the challenges you faced initially?
I'm not going to make this into a my story session, so you're not going to hear about some remarkable things which I did, not because I did very little. But what I'm saying is the time when this venture we are talking about is in the late 1980s. That is even before the economy opened up. 1991 is usually known as the time when this liberalization started. My venture started I commissioned my plant in 1987. Now, those were very different times in many ways. And it's not, 1991 was not as if we all woke up one day some day in 1991 and suddenly everybody said it's a new world. Actually, only today when you write about it, now people know that 1991 things changed. At that time, not much had changed. So the thing about technology startups and know-how based startups and high technology startups, especially in a field like in healthcare, faces certain problems which I think many promoters, many potential entrepreneurs here need to be very aware of. When you're dealing with healthcare, you're dealing with a very conservative medical community. And they're concerned, so using anything new is not something which they commonly do. You think that doctors and all that are very ready to use new things. As long as they're made abroad, they will use. If they're made here, they're not interested. Then they will find all the arguments against it, that it is risky, it is this, it is that. They ask questions which are never asked from a foreign product because the presumption is that the foreign company has done all this. I don't know why. Why is there a natural presumption? Of course, one of the things is regulatory. In the Western system, the biggest entry barrier to any business is regulatory. To qualify to sell your product, you have to meet very tough regulatory requirements, compliance. In India, you don't have to. So it's a very ironic kind of situation. So the one thing which we did right, to answer Dr. Ramchand's question, the one thing which we did right, and my advice to all of you is, even where you find a easier way suggested by somebody, not to break any rule, but there is a quicker way, a simpler way, etc., don't take it. That simpler, shorter, quicker way is never the best way. Take the tougher way, take the more difficult path, because ultimately in healthcare and in biological products, what you need is quality and safety and efficacy, and all these things have to be established. So even if you take a little longer time. When we entered I asked the Chitra Tirunal Institute people, what are the standards which I have to comply with? So then I was given the good news that there are no Indian standards. So normally a manufacturer will say that's very good news, because that means you can do what you want, and the Drugs Controller General has absolutely no idea about this product. Would you believe it? My license, manufacturing license for blood bags, this is the only blood bag manufacturing facility in India, and one of the handful in the whole world. My manufacturing license read, Penpal is licensed to manufacture anticoagulant citrate phosphate dextrose solution in plastic bags. So my license was for manufacturing the anticoagulant, not the blood bag, not the device. And the anticoagulant by value, complexity and difficulty was 0.1% of the value of the product. And that is what my license read. So I framed that license and put it up in my room, thinking, look at this kind of. So we were, that was, so this ignorance of the regulator, etc., could have been easily taken advantage of. And I could have kept putting out any product I want into the market, etc., hypothetically. But instead, I was sensible enough or foolish enough to listen to the Chitra Tirunal Institute director and head of research who told me, they said, okay, this is the good news that there is no Indian standard. Now, let me tell you which are the, so I asked them, which is the toughest standards in the world? So they said, it is the UK DHSS standards. They are the toughest. The product had to pass 194 tests and determinations before it's allowed into the market. I said, we'll take that. No need to do it. We took it. So today, I'm saying there is a connection between that decision and the fact that we are the largest manufacturer of blood bags in the world. We are the most efficient, most profitable manufacturer of blood bags in the world. We ship blood bags to the maximum number of countries, healthcare systems in the world. It's not an accident. It's because of that one decision we took. Everybody trusts us. The quality of our product is this. So it's a tough route to take, but if you take it, I think in the long term. So today, of course, compliance and statutory and regulatory, effectiveness is increasing. The regulators are getting smarter, they're getting more technically savvy and all that. So in every field, regulation is improving, which is good. So regulation is now, today, while you still come across people who tell you shortcuts of how to cut across, that conversation is coming down now. Now, even in board meetings and in management meetings, regulatory compliance is one agenda item. So the culture is changing, but this is the one point which I want to say. All the rest is the usual problems of startups, and being an ex-IAS officer did not help me at all. In fact, if I can say, at that time I was 30 years old. By the way, this is an error, because I can't write IAS after my name. I never write it. This is an error, because IAS is not a degree. It's a service, and I don't belong to that service. I resigned when I was 30 years old, after 6.5 years. So I don't get a pension, I don't get anything. Anyway, so it didn't help me, because many government offices where if by error, by something, if they knew that I am an ex-IAS officer, my problems used to start then. Because they didn't know how to deal with me. See, otherwise they know how to deal with you. They know what to ask for and this and that. Now they don't know how do you ask this guy for hafta, the usual money and all that. They didn't know all that. So I don't think it helped in any way, but other factors helped. And like I said, this decision was the single biggest factor. I hope that is of some relevance. Yeah. Thank you.
So I know that, the complexity of regulatory is unimaginable, especially novel, anything novel we are doing, drugs, biologics, etc., because I've gone through that, and eventually in India, hardly any phase 1. We are the first one to get some early approval for going for a clinical evaluation called what is called C6 in India for the novel biologics. And hardly anybody has got, or rather phase 1 is done. We expect to do a phase 1 trial, so I know the complexity. I know especially 1983, and especially if you're, I know that UK, the stringent, the regulatory requirements thing. So what we were thinking is that we will also ask some questions to students, and who is ready with the questions, please? No, you are not a student, but I expected the students to ask, but you can ask them. Anyway, so we can ask them.
Thanks. Thanks. Yeah, hello. Thanks, Dr. Ramchand, and the panelists for such a wonderful discussion today. I'm from CVJ Center for Synthetic Biology, myself Undi Krishnan, a principal scientist and head of the operations of the center. And we are focusing on, yeah, we are, we are the sender, you told me, yeah, told us, yeah. So this is, we are focusing on an industry-aligned academic program, along with strong R&D also. So, I mean, my question is that, way back in 1980s, you started, that's a deep tech that time. But, in 2025, because we were searching for, industries, biotech industries in Kerala. I mean, I was with Divya and also, other officials in the KSVM, Anub and all. So, but, we could not find any deep tech startups. Unfortunately, I don't know. I mean, probably we couldn't come across, or I don't know. We could not find any, a deep tech company, except, I mean, previously I was working with Cygenome's Alstonia Bio, which is a next generation antibody therapeutic evaluation startup, which previously I was working in US, and Cygenome has invited me to set up this facility. But apart from that, since, state has all the leading institutions like Sri Chitra, RJCB, and many of the leading universities, and thousands of students are passing out with extraordinary inventions, in fact, many of them. But all end up with kind of an RIP in some libraries, probably, I don't know. So why this is happening? That's, yeah.
I'll just give a very quick answer, and then after that, I think my much more knowledgeable people. One of the problems is the regulatory uncertainties regarding the regulatory approval process. They are not clear, they are not transparent. There is a big problem on the side of the regulatory, especially in the case of devices. I know. I cannot tell you about biologicals, but I know in the case of medical devices, because I used to be member of the Indian Standards Organization committees for this. So there was a lot of confusion there, and there is also turf battle between the Health Ministry and the Department of Science and Technology as to who will control. So under whom should the Medical Devices Regulatory Regulation Act, MDRA, as it was originally called, that finally has been resolved, in my opinion, in the wrong way. That's my personal view, by giving it to the Drugs Controller General of India, because they are all people coming from the pharmaceutical industry. They have no idea about engineering and technology as applied to devices. So this is a whole different class of products which requires a different kind of. So as a result, this delays in the approval process and ambiguity about how a proposal, how a new product or a new process should be approved is the valley of death today, where the startups are not able to get beyond. And some way has to be found for this. And actually the Medical Devices Regulation Act, which was being drafted, there was a committee of 2 or 3 people, outstanding people, one from IIT Delhi, one from Sri Chitra Tirunal Institute, and one from somewhere else. That committee had recommended actually that a kind of hybrid between the US model and the European model should be adopted. And the European model, they said particularly, third-party certifications should be accepted. The US thing is no third-party. Everything has to be done ab initio, de novo, and to their satisfaction. And actually many US device manufacturers have got their products approved in Europe, because they've got so fed up with the US system. It's extremely frustrating and bureaucratic it had become. So I think, my answer is that is possibly one of the reasons why, because no investor is willing to fund a startup, because this gap is a big question mark. It's like an induction gap. They don't know how he's going to jump across this gap.
Just in addition to that, and my experience of dealing with devices, first is that the regulatory landscape has, in India especially, has evolved quite a lot than what we used to see earlier. Food and Drugs Cosmetic Acts, which we all have heard of, which is very vague. We have come a long way. And we as the developer have gone through the transition to dealing with multiple bodies at New Delhi, starting from RCGMs and all the people who are basic scientists who have never done experiments in animal models evaluating your drug files. You go to the centers in RCGB and your files are stacked up where everyone can just pick it up and develop their own products. We've all gone through this. But now we have come a long way, honestly. Along the products you go and start dealing with these regulators, there is more of learnings by these people sitting in those offices with industry. There is, of course, a borrowing from understanding from different parts of the world, mainly the Western world, which is for better. There is a more involvement of the industry, academia in that sense. There are questions, something which happened recently, as recent as when the BIREC was established by Renu Sarup, was initiative by her. And she made this kind of a pitch to a lot of people, and I'm one of them, who said that whenever we used to go, said, you guys have a lot of knowledge. Why don't you kind of impart this and help us understanding what industry wants? And this is what exactly happened in the West. That there is a industry regulator dialogue which happens constantly at the FDA or EMA, where they get to know, and they say, please tell us what is the new technological advances happening in the industry, because you are at the forefront of it, right? So there's a lot happening. A classic example is the policies for advanced therapies. Now today, can you imagine, we have only 2 products in cell and gene therapy from India, one from ImmunoAct, another is from Immunil. And we already have a policy for cell and gene therapy. That's a very good advancement, I would say. At one point in time, we did have a biosimilar guideline. We all contributed to it when the document came, right? And it ultimately happened. Now there is a continuous improvement in that. We used to go to multiple bodies to get clinical trial approval. Now you have a separate body which actually vets out your protocols and tells you whether it is statistically powered, blah, 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 everything. And you are not at the mercy of someone who has no clinical experience. So there is a lot of improvement. Coming to devices, there's something to learn from the medical devices, which we have been a part of, which is basically an injector device. The pens and the pre-fill syringes and all that. And the concept of the device is the product. As soon as you make this kind of a concept, everything falls in place. Because as soon as you say, oh, no, a cartridge is going to go into a pen, and then both are going to get injected, and you immediately start treating both separately. That cartridge is a separate product because it has got drug into it, but this is something, some kind of a device which will go into it and just you puncture the skin to it. No, that's not the point, right? Because the injector or the pen injector is the one which is going to decide whether the doses are precise or not. So it's very, very intricate part of the product itself. So when you say device itself is a product, for example, and you get a combo approval, the whole scrutiny and the eye for it continuously changes. And it has happened in India as well. So there are good success stories in India as well. We have to do a lot of catching up. Cell and gene therapy policy is the classic, and I believe there is also a policy being made in the smart protein area, where you can contribute a lot in synthetic biology. I am aware of several discussions that people are looking at engage with synthetic biologists who were traditionally engaged for strain improvements classically in antibiotics era. Suddenly it has come back, wherein wild type strains are being utilized for various products, fragrances, process intermediates, and they have no idea how to look at these genetic manipulations. So there's a lot from synthetic biology side to offer to this entire smart protein area, which is a part of BioE3 policy, by the way. Okay, so that's another point to where you can contribute.
I would like to address that one in a different point of view, because I think that your question was in a general point of view regarding how the translation is not taking place in Kerala. Yeah. So the simple answer is that. You are talking about no translation taking place in India. India. Yeah. Kerala. Yeah. Yeah. The one important thing is that. Yeah. Yes. What I think is that so far we were smart enough in generating knowledge. and the depth of value that we give to the knowledge was with respect to publication or degree. That is the only depth that we used to know, we used to give to knowledge. We were not aware of this type of opportunities and possibilities. Now it is high time that we have to explore not the knowledge, but our wisdom to make use of the knowledge into a more value-added form. There comes the opportunity, number one. Number two is the real reason is that all these things are happening mainly in the academic institution where the person who is taking lead of such things are not having any industrial exposure. So they are living in a so-called hypothetical, theoretical world where enjoyment is happening by generating data, generating publication, and a degree, and consider that, assume that this is the end and this is a perfect one. So we need to transform that one. I think that is the real reason. And from our side also, we are trying to generate some models because possibly by October 2nd, we are going to initiate some new startup in different area. And we came here with our team members also. Like that, what we need is the generation of more entrepreneurs from our academic institution, especially from the faculty members. If they generate models within the organization only, the students will get a real exposure that this is possible. So we can motivate the student, that is there. But up to what level we can generate models within the academic institution, that really matters. And I think our wisdom should work out in that area to explore this new opportunity and to give new value to knowledge that.
Yeah.
You can,
Go ahead, go ahead. I'll go ahead.
Strong, intellectual property and business development support system within the academic system, so that the student can interact and because see, I've been in years. We have a technology which is multiple of that is being patented, and we have a strong. I've seen that it's a strong system where they look at whether the value proposition is really fitting into the commercialization and such kind of things. I think probably we need to have such kind of very soundproof system so that student can approach and they get into translated or whether even it is even for patenting itself they take multiple steps whether it is really value for patenting because it's you need to go on paying the patent money. So I think we need to have these kind of mechanisms in place, and I think this will empower students or researchers to translate their technology into commercial value.
So can I quickly add on to that? When we say that industry, academia interaction and all, this is not happening very actively really in the academic space. And also most of the research problems or whatever we are taking up is not considering the requirements of the industry. So that also is there. So I think that this way the faculty industry interaction should be there, and in the even starting from the curriculum design we should have active industry participation, then only that expectations of the industry can be met and we can have appropriate manpower generated. Yeah.
Yeah. Sorry if I may. Yes, I think I really I think you both hit the nail on the head, spot on. And to your point, I think the challenge really is people who are doing good science in the academia haven't ever developed products, right? And so the way you progress after that, so up to getting a publication, using the preliminary data to write a grant and get the next grant, hire more students, etc., has been, I think, perfected, right? So we have done it for so many years, we know the process. But I think where in India, in general, what I see across the country, I'm not so sure about Kerala, but I see this across, is I think the translation of opportunities that actually can be productized. And it may not be all, right? So the other problem is, as an academic researcher, I feel that the discovery that I made is now $1 billion product, right? I think there is so much to do from the time you actually have a discovery to even see whether it is developable or not, right? And I think that appreciation isn't there as an academic to me. I spent my first part of my life as an academic and then in the industry, right? And I think that the challenge is to really discern, be objective, and decide which leads that you may have in your lab are actually going to be translatable. And I think to your point, I think you made a very important point. What is the problem you're solving? It doesn't need to be very complicated, right? What problem does your discovery solve? Who is your customer? Okay, and what would it take to make that product? And what are they willing to pay for it, right? If these questions are answered, I think you will know whether it is worthy of pursuing or not, right? And also lots of patents that are there actually are not worthy of the paper they are on, right? So it is very, very easy to just, but you know, there are different strategies for that. Sometimes you may want to have that patent just to exclude somebody from using it, right? But it could be a part of a bigger portfolio of patents you might have. So I think the important thing is how do we bring these interesting research outcomes from academic labs to translatable solutions that are fundable, that can be made into products. And if we can actually solve that, then I think you will see much more of what you want to see, right? And I think it doesn't help, as you heard, when the regulatory environment is still evolving and is somewhat uncertain. It doesn't help in the growth at this juncture because we are still evolving, but we have come a long way, right? In the last 10 years, our bioeconomy has grown over 16-fold, right? And we are, as per our BioE3, you heard Dr. Ramchandran mention that we want to be 300 billion in 5 years. That's only twofold from where we are, right? So in 10 years, we have grown 16-fold, right? And I think it is all doable, but I think what is needed is all the students who are here, please look into your areas of work and ask the question, whatever I'm doing, is it really solving a problem? Or am I doing it just to publish another paper? Okay, because our time on this planet is limited, we want to use our time to do meaningful, impactful work. So you owe it to yourself to ask, is this what I want to spend the next 4 or 5 years or 10 years, whatever, in that lab doing? Is it worthy? And I think if you can do that, if your goal is not a publication, but solving a problem, and if this is going to happen across our country, we will become, I think, the startup capital of the world. That's.
Yeah, I would just like to add to what he just now said and what came from all these discussions. We are today now IP and compliance and all that is generally accepted by everybody. When I started out on business, I don't think there were too many people in the country who could expand what was IP. What did these two alphabets stand for? What I'm trying to say is, so the whole idea then was, in our case, for example, was to make something and put it out into the market. I mean, we were manufacturers. If we had a problem, we had to find a solution. So what I'm getting at is, our main product was blood bag systems and everything connected with blood bags. We had a lot of R&D going on in that. Based on the demand from our users, which were the blood transfusion services of hospitals, they used to give us some equipment saying, this particular equipment, which is supplied by one of the foreign manufacturers, is very difficult to get, very expensive, etc., etc. So they just told us, can you make it? So we used to take it and hand it over to our engineering team and say, they'll open it up, they'll come back and say, hey, this is not rocket science, very simple stuff, And why should it cost 5 lakhs? We can make it for 50,000 rupees, making a good profit. So what I'm trying to say is, just like everybody on this planet, including the biggest offenders are the Americans, by the way, and they go around preaching to the whole world, everybody learns by copying. Imitation is the first stage of technology development. And everybody's done it. When the Americans' big protest about China and all, you should have seen the Chinese scientists' responses to it with historical evidence about how the Americans stole the technology from the British, you know. But at that time it was okay, you know. So my point is this is going on all the time. And we also, our initial learnings were all by copying and by imitation. We opened up all the products of in our lab, one of the things which we used to invest in was what were our competitors making. And our competitors, there were no Indian competitor, all were foreign, all were MN, big giants. So we used to get all that. So we took one of the equipment and we learned all this and we started manufacturing them, and probably we were violating IP, I don't know. We never got sued by anybody, but we must have been violating all kinds of IP all over the place. We didn't lose any sleep over it. We went ahead with our business. Finally, we reached a stage where CII sent us a global tender issued by the Taiwan Red Cross. Okay? The Taiwan Red Cross wanted equipment for their blood transfusion services, and one of those was a tube sealer, dielectric tube sealer. It has to meet very stringent specifications for hermetic sealing of blood tubing. We were making that. We just thought as a learning experience, let's quote. So we quoted, and then we forgot about it. We thought, how are we going to get it? An unknown company in Kerala, in India, and quoting to Taiwan for an electronic product. Lo and behold, 1 month later, we get an communication. Those days, you had telex before the age of internet and emails, and also many of you will be wondering this real prehistoric stuff. So we get a telex message saying, the tender is awarded to you. 250 pieces of dielectric tabletop tube sealers to be supplied according to the specifications we gave. So we supplied. And I still remember, I must have been sitting through the night personally checking every box which we were putting, because suddenly we got very nervous thinking, oh my God, if this goes and if it fails, just imagine the mess everything will be, and it's going to Taiwan. So we sent it. After that, I think I must have been going regularly to the Paravangadi Ganapati temple and breaking, I don't know how many coconuts there, and month after month after month, and we thought, there will be all kinds of legal notices and everything coming. Nothing, silence from Taiwan. And then finally we forgot about it. Now look at this, years pass. After 4 years pass, we get another telex message. By then it was email, I think. So we get an email from the Taiwan Red Cross. It is our policy to replace equipment every 5 years. Your equipment has been working with us without any problem for the last 4.5 years. We are waiving tender requirements this year and are inviting you to quote again for another 250 pieces of that equipment. Just think about this. So my point was, what I'm trying to get at here is, how did we learn about how to make a tube sealer? By copying. We learned how to do it by copying. After that, when we wanted to improve it, we went to CDAC and we told them, how can we improve this product? How can we make it smaller? How can we make it lighter? How can we reduce the number of moving parts in that? How can we make it more cost efficient, etc. And so we asked all the right questions. They had the knowledge, CDAC people, and they advised. And between the two of us, we came up with a superb product. I have even now I'm out of business. I sold the company 10 years ago, but sometimes I go to hospitals and the doctors, when they know that I am the guy who started Penpal, they'll say, sir, I want to show you something. And they'll take me inside. And I'm astounded to find a 20-year-old equipment still working. Say like a platelet agitator incubator. The platelet cannot be stored like this. It has to be constantly agitated. 24/7 that equipment has to work. It cannot stop. 20-year-old platelet agitator working inside its incubator, because it has to be in exactly 20 to 22 degrees Celsius. Working without a problem. Silently working there. You can not even hear the click as it's moving up and down. We learned there's only one way to make many products is to do it. to actually make it. And if you are sitting, preparing research paper, of course it starts with research, but my point is a lot happens by tinkering and trial and error, which is why you must have a relationship with a manufacturer or a workshop or a tool, because only at, the traditional view of discovery and knowledge generation was that there was a location. Where did knowledge generation happen? In the universities, in the research labs. And what were factories? That was where knowledge was applied. All those kind of hermetic divisions and silos have gone. In fact, today it is a moot question as to whether more knowledge generation happens on the shop floor or not. I am of the opinion that for many engineered products, most of the knowledge generation actually happens on the shop floor, because they face problems. The whole line has come to a standstill. You have to find a solution, otherwise the line cannot start again. That's, like somebody said, the greatest creativity happens under the maximum constraints. So when you have a line which has stopped, 100 people are sitting idle, you have to find an answer. That's when you find real ingenuity, real innovation and real creativity and all happens. So what I'm trying to get at, I went about in a roundabout ways. See, this old-fashioned copying, imitation, please don't think these are something wrong, please something to be ashamed of, something to feel, you have to, nothing. Everybody did it. Everybody today who goes around preaching about various things, they started that way. And if we think we are going to get to there without doing this, we are seriously mistaken. Learning is by doing. Manufacturing, shop floor, in the front line, with the equipment, with the workers, with the technicians. If you're technically, scientifically trained, knowledgeable people, sit with them and work with them. That's where, at that intersection, you're going to find all these developments.
1 minute. So there is, I think, tea and something to eat also is waiting. We were supposed to have the tea by 3:30. So can we do one thing? Can we go have a tea and everybody can come back? Because a very interesting discussion is going on. Very interesting. And I wanted the students also to ask some questions. That has not happened. So if it is a very quick question or answer or comment.
Question. It's a quick question.
Yeah, please.
So I'm Dr. Arunabha Chakravarti, working in Semisco Biologics, which is being incubated in Genome here. I'm leading the antibody production, therapeutic antibody production purification facility there. So my question, so we talked about biosimilar and biobetter and the novel therapeutic antibody development, monoclonal or bispecific antibody, bispecific monoclonal antibody. So my question is, with respect to manufacturing efficiency and then maybe regulatory aspects and economic sustainability. So as a startup or a company with only a few years, so which domain you suggest to or advise to jump into so that the chance of success is much higher, considering all these three aspects of economic sustainability and regulatory aspects, as well as the manufacturing efficiency?
Maybe a clarification for everybody. I mean, I'm just asking this question. So, are you interested in developing novel biologics?
Yes, we are in that domain now.
Okay. Yeah. Are you into discovery or development?
We are into, design plus development.
Both discovery and development. Discovery also is there.
Yeah, so that's very challenging to decide, I think, right? Because if I look at examples of companies that in this space, in the antibody therapeutic space, people have, both are equally challenging, right? And both have inefficiencies in them. So the question you're asking is actually very complicated, because any inefficiency across this whole value stream is going to compromise your position. If you are more confident about discovery, if I were you, I would focus on doing that and making it the most efficient process that you can to discover high quality maps that are going to be very, very potent, right? And then possibly out-license them and then have some of your candidates, you don't out-license all, right? You invest then in development. If you're a scientist, right, I would focus on, or more of a, not a biochemical engineer, but a biology scientist, I would say focus on discovery first, try and get good molecules, and then try to out-license them, and then focus on some of those if you want to build your capabilities on development, which is again a long drawn out. So discovery is a shorter time, a lot of work, right? But development is going to take much longer because you have clinical, etc. So there also you may have to take a phased approach. If you are a startup, if you are well funded, money is not an issue, then it will be different. But that's my 2 cents.
Yeah, in continuation to your discussion, let me also, because I've developed 2 molecules, absolutely novel. And, I have like if you get time, I will discuss that, some of the things which you mentioned connected to regulatory, I disagree with that, but I'm not discussing that. The second thing is that you mentioned that out-licensing. So we have made up to everything. As I mentioned, the very first molecule, till date, no one has got a phase 1 approval for a novel biologics from India. But then we don't have money because after that, we have to go for phase 1 in India or US. So we went to US. Then for IST trial, investigator supported trial, there was some queries were asked. So we thought that we will go for out-licensing. Everyone wants after phase 1. That is the problem. Nobody, no company, because I met with Dilip Sanghvi. He told that why don't you complete phase 1? They also don't take that one. So that is the problem. And if you are able to, see, ours is like, not a very big, ekdam chota, very small company, somewhere put money and, came up to this level. I'm not telling that still we spend about $5, $6, $7 million each one. But then to go for phase 1 is, if you are going to do in US, it will be between $3 to $5 million. Nobody is giving. They are telling complete phase 1. Even the investors are telling, because they don't want to take risks. That is what everyone is telling now.
Yeah.
Yeah, we can. Why don't we do one thing? You can continue the same thing. Let's go for tea. Oh, okay. Yeah, we will continue the same thing. No problem. Because everyone is asking for that one. And please, students, ready for your questions. So we'll meet in 10 minutes. 10 minutes. So the discussion is going very interesting. So we should continue.
So can I see a show of hands? How many are students here?
We are students, please.
Okay.
Majority are students.
Great. Yeah. So look forward to some questions from you guys. Hear about what you're doing as well.
I request everyone to please be seated and we shall resume this interesting session. Yeah, let's start. I'll go out and call them. That's true.
I think few more. She will give. Pamenda. I think few more people have to come. Dr. Divya, you only will have to go and ask them, otherwise they will be going on drinking tea. So,
Meantime, I would like to make a comment on what Bal Gopal sir was saying. He was speaking about the copying of technology that we do here and get things done. I mean, I totally agree with you, sir, because I run a lab here which is sponsored by the Wellcome Trust and at Cochin University. So therein, so it was during COVID, we had this crunch. There was no money. We couldn't buy some of the equipments which I had initially proposed. So what happened is there was something called the RNA workbench that we needed to purchase. And the funding agency said, now let's not buy all that. You work with the BSL2 lab, the lab that you have. Just go ahead with that and do whatever you want with it. So, but I wanted an exclusive RNA workbench. So there was this uncle in the neighborhood of the university who used to weld things for us. So I went and told him, Uncle, this is a problem. I want an equipment such as this. And the photo that I showed to him was thermo RNA bench. If you come to my lab today, you'll see thermo replica sitting over there at one corner, which this particular uncle made for me. He's like maybe 60 years old, and he made this for me, a pretty good, professional looking, but without the thermo label on it. So we just did the dissection and made it work for us. Yeah, it's just a comment on what he was saying. Definitely.
wait for 2 more minutes, or because at least 15 more people have to come. Eh? They're all, okay. So, as I mentioned earlier, that students' question, you will have to ask some question, otherwise, it will not continue. So, let's, someone. Yeah, please. Great. Are you a student? Okay, that's all.
Hello, panelist. So my name is Karthik Unikrishnan. I'm the co-founder of Havnos Biomolex Private Limited. So I have like two doubts. I'm just narrowing down one question asked by the previous member. So if we are having a basic upstream downstream clean room facility, so if we are planning for picking a biosimilar, not the novel biologic, but a biosimilar for production as a startup, so we are nearly like 2 years after starting. So what are the criteria that we should consider for starting the production of a biosimilar?
Yeah, thanks for the question, and I didn't get your name of your company. Could you please? Okay. Thanks for the question. Clarification. When you say production, do you mean manufacturing or development, or where are you on the development? Okay. So you're asking how do I select a candidate molecule, or are you saying you have a?
So the question was not clear.
Yeah.
Are you in the, see, there is various phases are there. Discovery phase, development phase, then, the approval process, RCGM. There is C1, C2, C3, like, multiple approval process. Then subsequently, the most important thing is the CGMP manufacturing material generation for clinical evaluation. That is what one of the earlier I mentioned. It is very difficult because, it's not like synthesizing a small molecule because, the facility, and I will tell you, I have got 4 or 5, I'll just finish that, you will be clear. 4 or 5 codes to do material generation. And first is from Lonza, the other one is from Biocon. Just 1 gram, 1,000 vials. You know how much quoted? Lonza quoted $10 million. Biocon quoted $3.5 million. The other one is, the other company is there, like, because I know only 3 companies, $3.5 million. Just small this thing. And everyone wanted 3 to 4 years time. So, the only way, if you want to be, because I know by what I understand is that you are a small company, isn't it? Where you are now for this biosimilar. At what stage? Have you applied to RCGM approval, IBSC, plus...
Oh. Yeah, can I? Yeah, go ahead.
No, he has not yet started.
No, so. Yeah, that's what I just wanted to know. So your thinking is that you wanted to develop a biosimilar product, correct? And in the manufacturing mainly, or manufacturing development, process development, correct? Upstream, downstream formulation, which one to begin with? That is your question.
Yeah, so I just wanted to get that clarity so that, are you saying that you have a molecule that you want to develop the process for? Or are you saying you want to identify a paper exercise to identify which molecule you should develop? What's your question? Second one. Okay, so that's a more strategic question, right? And you want to, what's your end game? What do you want to do with it? Okay, a process for the product. Yeah, so okay. So, I think you'll have to really think through, as you heard, the whole process of developing a biosimilar is A, time consuming, B, expensive, right? You need to figure out your strategy at various stages, how you will fund it, right? But you'll have to start, you also have ahead of the process development, you also have cell line development, right? And you may want to look at what are the molecules. Because it is a biosimilar, right? You know that there are a number of molecules. You'll have to do an IP assessment for the landscape, right? Figure out what are the molecules that are going to come off patent by the time you will, or you will need to be able to develop it before the molecule gets off patent, right? Otherwise, your value will erode, right? So you need to think about the timing. When you look at that, you will get a number of potential candidates that you could pursue from the novel biologic space, novel antibody space. Let's keep it simple, right? Then you can ask, what are the indications for all these different molecules? What is the market size that it addresses, right? And then you can also look at what is going to be the degree of difficulty in making that molecule. You need to study those molecules and ask what is going to be the technical probability of success if you were to develop, given your team's capabilities, right? So that's important. This is if you want to develop it in-house. I'm talking about all this, right? Then all this applies. Then you will filter the molecules this way, right? You want to have the highest, a growing indication for which the market is going to grow, number one. Number two, high total addressable market that you would want to have, right? And three, you have to look at the technical fit with the capabilities, and look at all those patterns, look at the literature, look at what it would take for you to develop, and what's the technical probability of success that you will match that molecule. I think these would be the criteria to take into account. Even if it is not the top molecule in the list, let's say, for example, you have a top molecule in the list, but you don't have the technical capabilities, right? Then I would not go after that. Even if it is, let us say, only 200 million or 500 million today, by the time you actually make your molecule, if the indication is such that the demand is going to continue to grow, the same molecule will actually be multi-billion dollar. Okay, so, but the key is for you to actually have a biosimilar molecule on your hands. Otherwise, you don't have a game to play, right? So focus on your strengths, focus on, assess your capabilities to develop the molecule in the shortest possible time. And also look at another area to think about is also clinical trials. So for the molecule, again, that is changing right now. So, there is a bit of an uncertainty there. It is possible that by the time you actually get to that process might get shortened. The cost of development as a result could go down. The time to development could go down, but then the competition may go up because of all that, right? So you may want to look at that as well. Certain indications, the time may be high, cost may be high, you will not be able to put it in the market, right? So you may want to not pick molecules that will fall in that category. So you actually have to look at, on paper, because you already know the indication, the product, the novel biologic, you have to do a lot of paper exercise to start with to look at the molecules that you want to zero in and choose for your development. I hope I have addressed your question.
Have you worked in any of the pharma, biopharma, biotech company? Which one? I know that's what we have. So you might be knowing our molecule. We have developed our CRO was Levim. So Levim is making 2 or 3 molecules. Right. Yeah, please.
So, good afternoon, everyone. I am student of biotech, currently studying just completed my master's in NCH. So, my question was like, sir was discussing about glycoprotein. So, basically a chain of sugar which bind to protein. So, as per, I don't know much about, but I know few parts like E. coli naturally don't produce don't do glyco. Yeast used to do, but they are not good for non-humans. Mammalian cells used to do like CHO, HEC. These are used to do, but they are expensive. Now we are the age of AI and all. So these AI are good for predicting and all. They are efficient. Like company like Glycosi, now they are acquired by Merck. They have engineered engineered yeast glycosylation process. Google DeepMind, along with the Caspi protein, they develop like now into the glycosylation modeling and all. Another company like Evotech Biologics, they have driven AI-driven modeling, bioreactor. As sir said, like copying is not bad, sir was telling. So is it possible to integrate all the process like host cell state, bioreactor condition to accurately predict and control the exact glycoform and distribution of therapeutic protein before production? As sir already told, like it's very tough to accurately predict the protein that should bind the sugar. So this AI is like, AI also can do wrong thing, but their capability is more better than humans. So is there any chance if I want to do, in future, if I want to develop some company that is completely run by that, what will be its possibility? Thank you.
Yeah,
Just
but then definitely that, you know, if you want a glycosylation model to be there, there should be an algorithm should be there. So, I mean, I don't think, because AI also has to depend on the earlier some data which is developed. Otherwise, like, you know, see, the glycosylation process is not based on the configuration, but it is probably a non-enzymatic process, but it is based on kind of, you know, the glucose goes and binds to a particular place based on the what is called the steric hindrance, etc. So I feel that there is probably an algorithm should be there. Without that, you know, AI can't. That's what I feel.
Yeah, just let me just compliment you for your thought process. Okay, I'll let.
Yeah, so it's a doable concept. Let me tell you that, and a lot of people are doing this. Another someone mentioned about fudge display. Okay, I know people who have done these kind of AI-driven approaches before actually getting into a fudge display or creating libraries. So you are right. We have worked with Glycofi, by the way. Okay, and you can do predictability. Especially for proteins which are very, very heavily glycosylated, where permutation and combinations of glycosylations are multifold. Now, to know the impact of each glycosylation on the pharmacodynamic property is very difficult to predict, and design experiments which will take number of years together. So yes, there are approaches which people do. It's a very good concept, but Dr. Ramchand's view also is that you must look at the practical aspect of it. That is, what are the algorithms to predict such a concept to support it. So you need a good combination of a biologist and especially area of bioinformatics and someone who is good at, you know, AI. So I think you should continue with that. It's a good concept.
Absolutely. I completely really thank for your concept. It's a very good concept, and it is doable concept, and not only that doable concept, we are doing exactly whatever you have proposed or you have communicated, exactly same thing we are doing. But one part is very important. AI comes, only AI you can do if we have the data. Correct? Now, Biocon being an established company and working for multiple products for maybe decades, so they have quite a lot of internal data. Giving a specific example of the glycosylation, AI can be done in the same concept, can be applied in the different places, but the glycosylation example you have taken, we are just in the tea time, we are discussing with, I'm sorry, I missed your name, exactly the same thing. See, the glycosylation pattern can be modified based on your media composition itself. Correct? There are multiple other things. There are the molecular level, you can do that one. The cell line to cell line, so you can make a difference of the glycosylation pattern. So you have the data in hand, right? That means in this cell line, in this in this media composition, under this production condition, my glycosylation pattern is A, B, C, D, E. With a different permutation combination, I have a full set of data. Now you do AI. Correct? You do appropriate AI. You can be very close to whatever your requirement is or whatever you are looking for and maintain the process exactly in the same way. That is doable. And it is, and not only your data, there are multiple public domain data available. That also you incorporate and do the complete modeling. See, AI is a terminology, but ultimately it has to have from the data itself. So that's all.
That's what. So something like this. For example, if you are giving random amino acid sequence, which is not existing any protein till date, so whether AI will be able to predict the conformation or not?
Yeah, we have AlphaFold, which has.
No, what I'm telling you, absolutely random amino acid sequence. Absolutely random amino acid sequence, whether after putting it that, it is not connected to any sequence of existing protein. Whether AI will be able to predict the conformation, protein conformation or not. Okay. I think no.
It will be. No, so recent advances have actually, this was the concept that we had for a while, right? And there's been a lot of work that has happened. I would say last year or 2 years maybe it has been now that the algorithms to solve unnatural amino acid.
Provided the algorithm is there. That I agree.
It's there actually.
If it is
right
there,
now,
the 100%.
You can do that, but we need to verify, obviously.
That's what I was telling. Absolutely random amino acid sequence. Once it is given, let's say 1,000 amino acid, whether AI will be able to come out and tell this is the configuration already given, confirmation.
Yeah.
But it is going, because 1,000 amino acid, it is supposed to be having that many millions of interactions.
Long range, short range, all that.
Yeah, so it is, what I am telling is that, yes, provided every amino acid at least that, confirmation is given.
Yeah.
Then I am sure.
Yeah.
The same way,
I'm not
some
sure
kind of, what I feel is that if you are getting the existing glycosylation and the points by a particular method, which is already there, you will be able to predict with the AI.
So let me. Can you speak a little louder, please?
Oh, sorry.
So it was basically like ISKNV, till now there is no vaccine for this necrosis virus. So CERT already told me about AlphaFold. So use all bioinformatics server and all, I designed a particular protein sequence and checked through bioinformatic tools whether it will work or not. So I got the good result that it will work for the necrosis virus, and it was producing good amount of humoral and both type B cell immunity and T cell immunity, and it was showing good result. But sir is also correct. We have to check in the fish, live fish, because I have a protein sequence which is made through AI and bioinformatics tool, but we have to validate through that. But chances are high. During COVID time also, people have designed the sequence using that thing and validated. So similarly, we can do it. My question is, as sir told, like during that process I was designing, I was facing huge problem to gather the data, because there is no data available on the set, And if I want to get the data, I have to consult with. How can I means consult to that people give me data? Because
Have you tried to pitch this idea
yeah,
to any seed funding agency?
no,
You have an idea currently, but you're saying to get to the next level. How do I get to the next level?
so
How do I get to the data? If the data is available, there's a money requirement, whatever those things are, right?
so
I
no.
think
These things together is to approach your idea to a seed funding, the government or Bionest or
no, I think his question is different. Yeah.
Yeah,
His question is how
getting
do I
that data.
access?
there will be resources to get that.
Yeah, it's there.
You will require money for them,
Yeah.
right? So ultimately it will boil down to this, and then you have to approach the seeding agencies, okay? And there are seeding agencies. Pitch your idea to them, saying that this is what I would like to do.
Yeah.
I think today is the ecosystem where you will definitely get a seed funding.
Just I not tell you, just pass out my master's.
Yeah,
like,
yeah.
Uh,
It doesn't matter. You can, yeah.
yeah.
You would have heard the classic case of West Plan students who
Yeah.
got the order from army
Yeah.
to make a drone, right?
Yeah.
Yeah.
It doesn't matter where you are.
S-
It's about the idea.
Yeah. See, some of the biggest tech companies have been founded by college dropouts. So you are way more qualified, right? So. I'm saying you are way more qualified. Don't underestimate yourself, right? That's my point. Yeah.
So this was my curiosity question. I want to work. I want to learn more about it. And future, I definitely want to produce this thing. So thank you, sir, for.
Very good. Yeah. So thanks.
Thank you very much, but the last part I didn't hear. But if you want to know about reverse vaccinology, somebody I know in that university, I'll tell you, is in Gujarat. So they are going to Japan. So they are developing mainly so many the vaccine candidates based on this methodology. I can connect with you. So Marwadi University. Yeah.
So
I'll give my card so you can contact me.
just one clarification I wanted to give here, because you are mainly looking for data to make it algorithm, or maybe that's to get some information. Looking for the data. Data is the problem you are facing? Okay. See, guidance देने के लिए you will get lot of people. But to be very frank, this type of data is mainly available in two places. One is the company, and the second one is the academics who has done certain activities. In the company, don't expect that they will provide you the data. It will not be. Correct? So you have to rely on the academics. And second part is that there are public domain information. Some of the information will be available in the publication or maybe in the patent. So those are the things you have to track. Do not go by any company. Nobody will give you the data. I am very blunt and frankly telling.
Thank you, everybody. So you please sit here, continue to sit. We have 3 Ideathon is there. So I think really wanted to continue, and my questions also still 90% is like that only. So we can, I was just talking to Dr. Balgopal. He was telling such meetings or workshop or discussion should be there, especially about mainly about biologics and novel biologics and similar things, and definitely he will support in whichever way he wants. So I think I'm suggesting Dr. Divya to what he was telling is that we should continue, and I told him to also to discuss briefly tell about this. After that we will start with the Ideathon. So our guests will sit here. Yeah. So what I'm telling is that what he has suggested, he is going to tell that. What are, I don't have anything to add. Yeah, okay. So basically this looks to be very good, very happy. All the panelists seems to be very happy with the questions and the response from the students and the startups. So you can. Yeah, they can sit here. Yeah. So you can sit there or no? Okay. So there's a memento to be given. So, Divya, Dr. Balagopal will have to go, so you can give the. Yeah, you give it to everybody. Yeah. Yeah.
Now we come to a very important moment of this session. We would like to present, as a token of our appreciation to all the panel members, a few mementos. I would like to call Dr. Divya Purushottaman, our facility manager of Cribs Bionist, to please do the honors. Yeah, okay. So first we would like to present our memento to Dr. C. Balagopal, sir. Very inspiring stories. Next, I would like to call Dr. Nitish Dev. Dave, sorry. Next, Dr. Partha P. Hazra. Dr. Radhakrishna Nike. Dr. Gopala K. Dasika. And finally, but not the last, the only female panel member, Dr. Valsamma Joseph, ma'am. And I'm honored to also call upon Dr. C. N. Ramchand, sir, to do such a beautiful session over here, that we have beautiful ideas. Yeah. A round of applause for Dr. Divya. Divya, the Ideathon. Name, yeah, it's here. So let's call Divya Jose first. Okay, so for today's Ideathon session, we will call Dr. Divya Jose to present her idea.
So good evening, everyone. I'm Dr. Divya, working as a postdoctoral fellow at NCH QSAT. Along with that, I'm running a startup company named Microsymes Private Limited. So as a part of my... No, the PowerPoint slide is displayed there. So the product what we have developed here is the nanocellulose fibrils that has been isolated from coir pith. So nanocellulose fibrils has got variety of applications, such as it can be used in drug delivery system, it can be used in tissue engineering, and also in waste treatment. So what we have developed is a powdered form of nanocellulose which has been derived from coir pith. And we have characterized the material. It has got a couple of application. So we have done up to the laboratory scale production. So the next step is to take up into the commercial level. So the major constraint is with the financial constraint of how to take up to the commercial level. So No, no. Actually, in the laboratory scale, we have tried in environmental bioremediation. Right now, we are trying to solve the contamination of effluent treatment plant. Like we have immobilized a bacterial consortium into the cellulose nanofibrils so that it can be applied into ETP plants. That is one area. Second, these nanocellulose fibrils as such can be used as flocculants for the settlement of organic matter. Cellulose fibrils, which, yeah, it has got a couple of applications. I've just pointed out at that. So the major next step is to take up into the commercial level. Actually, now we have tried with the chemical process like acid-alkali treatment. It is the raw coir pith. Raw coir pith. From that we have to remove the lignin and hemicellulose. No, this particular nanocellulose has got higher surface area. So the what you have tried is the bioremediation by attachment of bacterial cells. So nanocellulose has got higher surface area, so bacteria will get a higher surface for attachment, and it will improve the shelf life and stability of the product. With the no, it's a powdered form. Yeah. Simply put to the ETP plant. Like by 1 liter of effluent, you have to calculate how much grams it has to be added. Usually chemical treatments are done. And this is a biopolymer on which natural bacterial consortia has been attached. It is purely a biological product. Yeah, a lot of bioremediation products. Some are using chemical ones, some liquid microbial formulations have been used, and in some ETP plants they have been equipped with some bioreactors.
The basic question is that, what exactly it is replacing? Because when you are making something, so many different types of cellulose is there. Cellulose, hemicellulose, and hydroxymethane, hydroxypropyne, various things for various applications.
Yeah.
So when you are going to make something, there should be an absolutely novel use, which is otherwise not there. What will happen is that, if you are making something similar already, you will find it very difficult to commercialize.
Yeah, but still.
So first of all, you have to see, let's assume that, this nanocellulose you are making, it will be much cheaper than any other existing one. Let's say an existing kind of cellulosic material, which is, let's say, 10,000 rupees per kg or 5,000 rupees. Now you are telling nowhere you can get, and you can make it for 1,000 rupees and sell it for 2,000. Yes, there is a possibility. Otherwise, it will become very, very, very difficult.
Yes.
I always tell an example of, have you seen another equivalent paracetamol? It will not come. There is no fun in making it. Because it is very difficult. This is already an existing one. There is hardly any problem with paracetamol. Nobody will buy yours.
Yeah. So, but nanocellulose fibrils, it is not that much utilized in the waste treatment market, and still there is need for this type of product in any industries can utilize in their ETP plants.
Nanocellulose is used in pharma in the formulation.
Yeah, that is another aspect.
Right. Now, what is, have you ever compared with that particular?
And actually, we have just developed into the laboratory scale. Pilot scale studies has to be done.
Let me tell you, what you have to do is that when you are making this, the first thing you have to check is there going to be
Viable in them.
Yeah, a consumer is going to be there. Somebody is asking us some product with this thing. But I don't think this nanocellulose and all is like all these different types of cellulose already, there are so many manufacturers, so many applications are there. So first you will have to tell what is this compared to the existing. So
idea of pitch, basically, couple of things are required, because idea what exactly you are looking for, right? So first of all, what is the problem statement your invention or something is addressing? That is one. What are the comparators available against which your product is better than that? If at all there is a cost advantage, then that must be also brought into it. Okay? What is the cost of the current technologies and everything? That is second. And what kind of funding in amount wise you are looking for? Okay? Comparing that, attaching that with the little bit of a finance part of it. Because you are asking for money.
Okay.
Right? Money is for what? Even a small child asks you to buy, you ask them, what do you want for? Right? Same thing is for the investment. So that is how an idea probably should be pitched with a very clear understanding of what exactly your product is, what are you want to use it, how it is better than what is existing one, the money where it will be spent.
And also one more, very, very succinctly said, one more thing to think about when you are doing this is also what prevents someone else from doing it. How can you ensure that you are the only one with this kind of product? If all those are met, the next question that you also need to think about now is
Patents.
what will prevent me from doing what you're doing? Otherwise, you have no business.
You will never get a product patent for any natural product. But you
If
will get a
success.
process patent. And any process patent can very easily sum it up. So and that's not the main thing, but the first thing is that who is going to buy and use it? And this is the first thing you will learn.
Okay. Okay, thank you.
I would like to thank all the panel members for giving Divya a lot of ideas. It gave me also a lot of ideas to come up with. Okay, next we'd like to call upon the next participant is Adira. Ilya. Next is Arya. Karthik. Next person is Karthik.
Good afternoon, everyone. Good afternoon, everyone. Myself, Karthik Unikrishnan from Havnos Biometrics Private Limited. We are a startup incubated in Kusat TBI in Kalamasheri, and we are registered from Palakkad. So, this is the idea that we are pitching here. So this is called as, this is one of our product, which is called as CELLI, that is Cognitive Engine for Living Laboratory Integration. Now everyone knows what is a biofoundry and how a biofoundry functions. So this is a biofoundry concept that is specifically made to synthesize cell factories for expressing certain molecules. So this is a very basic concept of our product. This is the first line in our product. So, and the introduction, like synthetic biology needs fast, reproducible work precision. So we know that the traditional methods are very time consuming. So this CELLI is fully powered by AI, developed using a Python algorithm. And the idea is, it's a AI powered biofoundry working through AI integrated iterative loop. And it has a robotics, a specific particular type of robotics called as soft robotics, and databases and AI for automation. And it works on predictive modeling where the errors can be lowered. And the problem existing in is the manual workflows as well as the heavy cost and no real-time learning slows optimization. And the problem also with the biocontainment remains challenges. So the thing is that what CELLI does is that since it's the primary product, we can only develop up to 30 kilodalton protein, and that doesn't require PTM, post-translational modifications. So the database is curated only to deal with certain proteins that doesn't demands PTM, and we have designed an expression vector, a Vibrio-based expression vector that is that's very fast growing, as well as that can only synthesize proteins up to 30 kilodalton, and as in it's like, we know like it's since it's a prokaryote, we can't perform PTM using that. So our program like CELI has been shortlisted under the DBT BIRAC joint call for proposals on BioAI for establishing hubs under BioE3 policy. So also we have been, since we are shortlisted for that, we had a interaction with Dr. Rajesh Gokhale and Jitendra Kumar regarding our concept, and we are into the final stage of the BioE3 call for BioA. So that's with the concept. Sir, currently the expression vectors, the most powerful expression vector is done using E. coli. But there are strains of Vibrio that is like much more fast replicating than E. coli. And we are also addressing one particular problem, is that since E. coli is a terrestrial life form, we also found that there are lots of protein that is found in high saline conditions, like brackish water as well as marine environments, that is having problem with expressing in E. coli because of the salinity induced stress that is actually dealing with hampering the protein folding nature. So whatever we are doing this, once the protein is produced, the bioactivity of the protein is not retained in E. coli for certain marine and brackish water proteins. So that problem can be corrected using a Vibrio, and we have an expression vector that is the strain is it's basically a high saline strain. So, and it's the actually one of the most it is the fastest replicating bacteria in the world right now. So
Fastest replicating is okay, but in terms of productivity, how does it compare?
Productivity, like for the time being, like we have tried it with expressing cry proteins. Like we have tried it with expressing like different types of cry proteins, and we found that comparing the yield of an already existing what is that the E. coli expression vector, I think everyone knows, like BL21. That's one of the expression vector, commercially available expression vector. So comparing to the expression vector BL21 and this Vibrio strain, particular Vibrio strain, we found that the yield of cry protein is comparatively high. The production yield, when we analyze in the SPLC and all, the concentration and the bioactivity seemed higher than the E. coli vector. So the process here, what happens is that the whole synthetic biology workflow happens inside this biofoundry cabinet, and it's a primary design. So the thing is that it's only for that, only for the expression of this particular protein in Vibrio strain. So it's, since the databases is limited, and we have some troubles with this AI integration also, because we have like funding limitations. So now, since we have approached BioE3, since this BioE3 call was open, we applied this program in BioE3 BioA call, and we got shortlisted. Out of 443 applicants, only 12 got shortlisted, and we are one of that. So we are at the final round of the program. So we are submitting some documents for that. So I think.
So is this a grass organism?
Is this?
Generally regarded as safe organism?
Yeah, it's completely safe. It's a BSL basically 1. Yeah.
So you are making an engineered cell lines or an engineered protein for therapeutic or some application?
This is basically a bridge. It's basically a bridging process. If one of our client, the process stays with us. The thing is that if you have a PCR product, that is a single gene based protein expression, and if you have a PCR product, in case of bioprospecting, you have found some novel biological molecule, and extracting and testing the biological molecule, the concentration will be very low if you are extracting from the native world. That's why we went for recombinant DNA technology and expression. So if you actually can extract the PCR product of the biologic, then we can bridge the gap between the production as well as the expression. So that gap we are filling using our the CELI. So we are gathering the help of the AI for the process. So once we get the PCR product, we will start our workflow where we will optimize our strain to express that particular biologic in the system. So that's the bridge we are gapping right now. The thing is that the automation part and the process we have not yet started. We have only done it in the normal lab conditions. So we have tried it with E. coli for a comparative analysis between our strains, but not in the automation system. So that is yet to develop. We are developing each and every part right now. Yeah. Yeah.
So more applications you have with the known, it's a good starting point, no doubt about it, because you have its fastest growing and quick results and everything. But till you get to a stage where you really people declare it as a grass rather than you declare it, it's a huge kind of a convincing curve. It's a potential barrier, entry barrier for people to even trying out.
Okay, fine.
Because ultimately, you would like this to be get commercialized.
Yeah.
Right. So people should be using Vibrio system to use your stuff.
Yes, yes, yes.
And you must have the data to power AI.
Yeah.
With the known systems first.
Yeah.
As he said, for known systems you require data. There is tons of data for E. coli, very little data for Vibrio. So it's a good start, and it's a good initial idea. But to get to an application, it has to pass that muster of applicability and acceptance from the current norm.
Okay, fine.
The cell line you are making is better than the existing one. That's the problem. Second thing is that you have to go through a series of, just like a drug discovery process. This is the FDA is not going to approve this, but they want to make sure that nothing is excreted. Like
safety profile of Vibrio is, as you can imagine, it's I don't know if it's documented even. Right?
So is there any product? A couple of questions I have.
Yeah, sure.
Because this concept is very good. Conceptually it is very good, but you have to fill certain gaps.
Yeah.
To make you going to the next stage.
Yeah, sure.
All has been discussed, but couple of things. First of all, question: why you are not able to go beyond 30 kDa?
30 kDa is the limit for Vibrio. I mean the strain that we selected. So based on the literature and certain tests that we have done so far, like we were not able to express certain proteins beyond the 30 kilodalton limit. Yeah. We done it in the wet lab first. So from that only we came to this AA part. And regarding this E. coli also, like because of the server limit and because of the databases limit.
Yeah, you're right. So my question is, because something is not happening, you have chosen Vibrio?
No.
Or it is a logical step of choosing Vibrio for certain reasons?
Yeah, we have scientifically, we have evidence that for some marine proteins that is present high salinity, using E. coli or any other terrestrial strains is not an option, since some of the proteins have salinity stress on them.
I understand that, but how many people will come to you for such proteins that has got high salinity applications to try Vibrio?
So
You have tons of customers coming to you to use your factories or foundries at the micro or whatever level.
Yeah.
In available space where there is a huge opportunity.
Yeah.
Where we are looking at extremely low. So as we say, the concept-wise, to start something new is a novel concept.
It's a very good concept. But a couple of things I also just want to add. See, I'm interested in your concept. That's why I don't take it in a.
Sure.
Some of the gaps we have to fill.
Okay.
First of all, to go to any sort of new cell line, correct? Vibrio. I do not know whether any product is already available in the market coming through Vibrio. I do not know. No, right? Most likely not. If it is not, then to establish and to acceptability will be extremely difficult. I can tell you.
Yeah.
The reason
The only Vibrio people have the site of is Vibrio cholerae. Though there is a completely different, this is genus and species together.
Yeah.
But that's the only connection people can make, right? So, same kind of the reason is that legacy genes available in the same. And the safety profile is always question. Always question. There will be perceived risks. Any company will perceive as a risk and complete unknown area. They are territory. They are reaching. Okay. Still they might get it, but what vis-a-vis they are getting the benefit in going to it. You are telling that your multiplication time is very less.
Yeah.
Correct? Less means less than 30 minutes?
Yes.
How much?
The strain that we are using right now, the replication time is 10 minutes.
I give you the practical situation.
Yeah.
Because I work in the practical thing. How is actually? No, I tell you. Bacteria, 30 minutes, which is 30 minutes, right? Bacteria production in the fermentation. It's all in a day. It is finished in a day. It is never been a challenge anywhere. Yeah. Half a day. So and half day will be making maybe 1/3 day. You will not get any benefit. Or maybe that benefit will not reflect to take such a big risk to go a novel organism. That way, it is not balanced.
Yeah. So let me give you some more points.
I just want to add one more point.
Okay.
If it gives a very high productivity compared to very high it has to compared to the bacteria, then it some plus point.
Yeah.
Plus you will get. So those type of things here.
There is one huge advantage. So I will anyway tell the name. Yeah, definitely. So the strain name is Vibrio natrigens. So this natrigens is considered as the most fastest growing living organism, living bacteria. And it's a BSL-1 organism. It's not causing any kind of infections. And the reason why we selected this organism, since this is to support drug discovery platforms. So that's the thing. Discovery platform.
Instead of 100 molecules cloning and make some product out of it, I understand that will be a better challenge. I tell you whatever will be more acceptable. Drug discovery may provided a cloning, sequencing, expression. Correct?
Yeah.
In a normal way, in the E. coli, it takes say 3 months or 4 months.
Yeah.
But with your procedure, with a screening of 100 clone in your expression system, instead of the 3 months, it takes 1 month.
Yeah.
It happens. For the discovery, it is very beneficial, I can tell you. Doesn't matter whether it's a Vibrio or whatever, Vibrio cholerae also, it doesn't matter provided it makes some product. Because product, whatever way it is coming, it doesn't matter for the discovery. And that the proof of concept of the product is most important. Once the proof of concept is made through your channel, if you shorten the time of that stage, if the proof of concept is good, then they can change the experiment, they can go back to the E. coli for the actual process.
All right.
So the discovery, I agree, provided 3 months is becoming 1.
Yes, sir.
Then it will be a good year.
Yes.
You pitch that way also.
Okay.
So your target people who will do this is the early researchers.
Yes.
Researchers.
Yes.
They want to understand pathways. They want to understand how the system works. They don't have time. They want to crunch it. They want to do in a high throughput mode.
High throughput. Yeah.
Those people are your target people for just to get the data, publications, if at all patents, blah blah. The proof of concept establishment at the earliest.
Yeah.
That's the area, is your area of application.
Yeah. There is one more advantage with this strain. So the transformation process, like when we are inducing some gene for expression, Vibrio natrigens, this strain can actually take up genes from its media itself. So it's proven. Yeah, it can take genes from the external environment, because when we come to E. coli and BL21 and all, we have to do strict transformation process. So enzyme-based transformation process. But here we have an option. The thing is that there is a protocol that's already available. It's open source. So where they have done like transformation just from media, they just added genes PCR product to media, and they made this strain transform. So we have, when we are using E. coli, we have that problem that we have go with strictly like enzyme-based approach or CRISPR like that. So this is one of the advantage we are having a strain, that is yeah. 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 Yeah, definitely. Yeah, sure, sir. Okay. We'll obviously start with prototyping. Start with the prototyping process, as well as we need to do it in an automation way. So that's what we are doing right now. The complete process, the complete workflow should be automated. And it should be a proper biofoundry that is afforded by like medium to large scale labs. Like not like, I don't think the cost of our product will be too low. It will be some, we have estimated one range that not all labs could be afforded, but mainly since now the Biofoundry platform is available only at few labs, like more like sophisticated lab. But our system, we can be implemented in any kind of drug discovery platform. That's what we hope for. But again, we are on the research on that. So with the money, with the grant in our hands, like we start with the research, of course. Yeah. Thanks. Sure, sir.
So we can go with one more. Last one. We have one more participant. Adira.
Good evening, everyone. Myself, Dr. Adira Imdas. Actually, I'm an Ayurveda doctor. I'm something different from all of you, I guess. Basically, I am here to present some idea about Ayurvedic hydrogel eye patch. So I am treating mainly the eye disorders, and I found that so many patients are having the conditions like dryness, eye fatigue due to some digital eye strain like that. So, so the treatments in Ayurveda is like its setup is needed, and it's a long procedure, and doctor's suggestion, the medicine, everything is needed, and it is not much cost effective also. So I would like to invent some solution like Ayurvedic hydrogel eye patch. The hydrogel which is infused with some Ayurvedic medicine, and it can be used without any setup or like any therapies is not needed, nothing is needed, and it relieves the strain, eye strain, dryness, fatigue within 15 to 20 minutes of use. Like that, I would like to invent the formulation, and I have conducted the market validation, everything. It's like the digital eye strain, everything. It's like 500 million of people is affected with this eye strain, and about 62% in Indian cities. Indian also, it is affected with the digital eye strain. So the market size is very huge, and the competitive thing is like so many Blu-ray cut glasses and some eye drops, everything is available in the market, but those are not permanent. It is giving only temporary relief only. So this particular medicine infused hydrogel eye patch, it will give the rejuvenation and the therapeutic value to the eyes also. So that's what I am trying to invent this thing. I'm focused now.
So what is your comparative? Comparators, as we know, there are CMC-based eye drops, people use it.
Yeah.
The second one is the hyaluronic-based hydrogels are available.
Yeah.
As compared to that Ayurvedic, how do you compare the performance?
Actually, this will rejuvenate the eye protective mechanism. It will act upon the muscular system, the circulation. Ayurveda, the concept is something different. Actually, we will treat with the concept like Vada, Pitta, Kapha, like that only. In that, the dryness, it is affected because of the Vada, and like that, so many components we are taking for. So for that, the medicine I have taken, which will particularly act only for the dryness and for the eye rejuvenation, like that. Only the topical application over the closed eyes. Over the closed eyes.
As compared to drops.
Yeah.
Over the closed eyes.
Over the closed eyes, and which will be like some ghee medicine also I will infuse in that hydrogel.
So whatever the active ingredient
Yeah.
passes through the epidermis or dermis
Yeah.
layer and then acts.
Yeah.
That is what you're saying?
Yeah, yeah. Actually, in Ayurveda, the concept is something different. We have having some purambada, some external labor application and all. And this is also like the medicine, the raw material, the setup, the doctors, everything it's needed for those things. With that, with my experience and my knowledge, I have selected those medicine, and I am extracting those medicine and infusing to the hydrogel, and just placing it over the closed eyes for 15 to 20 minutes.
You looked at local irritability or whatever.
It's ideation stage only, and just only I'm initiating. Yeah.
I will tell you what exactly you can do.
Yeah.
So the first thing is that this doesn't come under traditional Ayurvedic. Okay, this is
Yeah.
what is called proprietary.
Proprietary. Yeah, yeah.
So if it is proprietary, what you can do is that you will have to get the approval from Ayush,
Yeah.
which is almost similar to DCGI because it is intervention.
Yeah.
Because something will go inside the eye.
Okay.
What you can do is that, and all the ingredients or extracts what you are taking is kind of known things, isn't it?
Yeah. Mostly it's like known things, but
there is something called, reverse pharmacology. In which what you can do is that you can get the approval from the ethics committee and you can use it and see whether it is working.
Okay.
So that will, instead of going to Ayush and getting all the, because they will ask you to do all the tox studies. So all the tox studies means not like a drug molecule, but the 28 days tox study you will have to do. You may not have to do any pharmacokinetics. You may not have to do any efficacy study, but a couple of tox studies you will have to do.
Okay.
Then you will have to apply for, just like IND with all those details, and they will give an approval.
Okay.
Instead of that, what you do is that because all the ingredients are known, isn't it?
Mostly it's known. Something
It is a known.
Yeah.
And for example, if the plant extract is like traditionally what you are taking, or if it's a known thing, but let's assume that curcumin is there. You don't have to worry.
Okay.
Phosphoric acid is there. Hydroxycyclic acid, these type of things are there. You don't have to worry.
Okay.
Because it's already known.
Okay.
So what you do is that don't do anything. First thing is that get an ethics committee approval and do this in 10-20 patients.
Okay.
Who has tried this? Have you tried it?
But the base material is not suitable actually. The stability is not getting. I have some.
You made something.
Yeah.
Had you tried it on yourself, saying that, oh, I have this strain, let me put it, or someone who
Yeah,
has
in
it,
some of my patient, I have. In some of my patient. Yeah. Yeah. Yeah.
It is working.
Yeah, it is
What
working.
you do is that do a reverse pharmacology approach. Then that is what your this thing, and if you want funding, you will get that based on this.
But ethics committee is a very good idea, actually.
Yeah, that is
To quickly check it. Yeah, because
so they will approve.
Yeah,
They will approve because provided these materials are known.
yeah.
Yes, now it was not there earlier. See, there are under Ayush, Ayurvedic system, if it is they are following something traditional, you don't need to do anything. You just get the license, just like a generic system. But something novel is there, they will ask for IND submission. So this definitely will do because it is intervention.
Yeah.
Because something will go into the eye, it can go into the bloodstream also. So what I suggest is that you go to the ethics committee, get the approval, and do in 20, 30, 40 people. The data will be like, you will be able to get. First you do a longitudinal study, then you compare with a placebo. I don't know whether they will agree for the placebo, but the longitudinal study they will agree. Then, once you have the results.
How do you assess the endpoints? Is it just a feeling of feeling relaxed?
No.
What is the tangible outcome?
Actually, some tests like Schimmer's test, everything is there for analyzing the dryness and for the comfortability of the patient also. They are having the symptoms also. Now the grading also we are having.
The grading system
With it.
is very good.
Yeah.
I think this will be a very good workable thing.
Yeah, it is true. It is very effective.
Yeah.
This will be a workable thing.
Okay.
This is something can be translated.
Okay.
And if you
Yes.
have any question for application, I am there.
Everyone needs that.
Yeah, here also most of the are wearing glasses and all. No?
So this is only for like stress and fatigue and dryness, right? You can't
Mainly for the digitalized train I am focusing.
Then I don't know how you can get a patent out of it. So if you have to get a patent, I will tell you what you can do that.
Okay.
It will be kind of composition, not a product. Composition, medical device, application.
Actually, I have applied for provisional patent for that proprietary medicine. In different multimodular form, I can apply. Like that, I have applied for the provisional patent.
Well, then when if you apply in 12 months time,
Yeah.
what I suggest is generate the data.
Okay.
As much as possible, and you will be able to commercialize this. This is a very simple thing.
Yeah.
But eventually you have to go to ICE to get the approval.
Okay.
But tangible outcomes are needed. Measurable outcomes.
Yeah, yeah, yeah.
The tests which are required, which qualifies it for releasing of the eye stress or dryness or whatever it is, has to be measured with the qualified tests to prove between any known comparator. If CMC ones are not better, if the hyaluronic based eye solutions are better, then compare with them under these parameters. How is this better? Is it equivalent? That you have to find out.
Okay. Okay.
Or not inferior.
Also, I don't know, sir, does it not require any understanding about how toxic it is? Is there any
When you go to ethics committee, there is because if it's a very well known product, because we don't know what exactly. That's why exactly.
He was saying also, he has
It is a very well known. Otherwise, ethics committee is asked to do a 28-day toxicity, and they might ask them to do an oral toxicity or an external toxicity for allergen toxicity, those type of things. They may not ask also. Because we have example like couple of the similar things. They have not asked the toxicity. It's a very well known thing.
Okay.
So I think you should go immediately. Then after that you go for this. It will be very difficult to get. You can get Ayush funding.
Okay, I have applied for KSUM also. Idea stage funding
The
is
most
also.
important thing is that filing and protection.
Yeah.
The patent and clinical evaluation. Initially as a kind of, as I mentioned, a proof of concept study. Then after that you can.
Okay, sir.
It can be introduced in a year's time.
Okay.
What I suggest is that go to some after protecting. There are so many companies out there, big companies. They will be able to.
Ayurvedic companies like Himalayan.
Those type of things.
I have approached them for third-party manufacturing.
So unless you have the data,
Okay.
nobody will. So go with the data.
Okay.
I mean, this will not have any problem in this area.
Okay.
Definitely this will not.
Okay. Okay, sir.
Simple idea.
Yeah, it's just a simple idea.
Good, thank you.
Yeah. Yeah, now the panel members would have a small discussion about selecting a winner from our participants, from Ideathon participants.
Okay. Is it? Okay. So do we need to? Or can we just say plus A? No? Yeah, I don't want that. I don't want that. Okay. Is this on? Can you guys hear me? It's on? Yeah, we can I have a now for the yeah. I think it's on. Yeah, it's on. Can you guys hear me? Yeah. So 3 innovative ideas, 3 presentations, and you saw how the panel here had really questioned and cross-questioned and tried to. The idea was to try and provide inputs that will help further the programs. All very good, you know, and I think always there is room for improvement, but I think all the 3, Divya, Karthik, and Atira, made good presentations. Wish you all the best. Hope you all continue to work on your ideas and take the inputs that you got today to translate it to a more successful outcome. But we have to do this job of ranking the 3 presentations, right? And I have been entrusted with that job. We did a collective effort here, thanks to all the panelists for coming up with the ratings. And any guesses? Who's going to be first from the audience? First? Okay. Yeah, that was very good. And I think that it's a great idea, good from a translation point of view. So the panelists actually looked at so many different aspects, right? And I think the idea is that all ideas are good. They also need to be translatable, and they also need to be successful from a business standpoint. At least the potential should be there, right? And we are not, you know, don't take our word as final. This is just our assessment today, right? And if you are not the first or the second, don't be disheartened, right? Because maybe a different panel would have given a different rating. So for whatever it is worth, I'll just give you the ratings. We all felt that Karthik is first, and closely behind is Atira. Both of them did a great job, but we had to pick one. I think there is room. There's more to be done, as we discussed, and you hopefully got inputs from the expert panelists here, and you will start thinking about how can you elevate the offering that you have to cater to a much larger market, right? And then the third is Divya. Divya, I think, I hope you got good feedback today. Did a great job. My compliments for the effort, but we all start somewhere, and we need to continue to believe in ourselves and continue to learn. So I hope you will take this as a success, that you came up, you spoke about your innovation in front of everybody, and you got some very useful feedback. So thank you very much for all the 3 participants to come up and speak to us about your inventions. Wish you much success going forward. Thank you.
Thank you, sir. And thank you to all the panel members for giving great idea and advices to the participants. Now I would like to call Dr. Divya for a formal vote of thanks. Yeah,
Okay, thank you. So, actually, it's a great pleasure to stand here and share a few words of thanks with you when we close this, because we started this in such a short time. And there was not enough time given to anybody, including the panelists, to prepare for anything. So I'm really happy that it went well. At least from where I look, it was a success. I hope you all feel the same way. So it was told before by our anchor, this program was organized by RGCB in collaboration with the CRIPS Bionest, basically to celebrate the 1-year anniversary of the launch of BioE3 program by the Government of India. So the DBT Institute is organizing a series of events for 2 weeks called BioE3 Pakwada, and this was RGCB's effort towards that. So on behalf of BRIC RGCB and CRIPS Bionest, I would like to start by thanking our chief guest, who is not here now, Dr. C. Balagopal, for inaugurating the session and also joining our panel discussion. I would also like to thank our all expert panelists who came in such short notice and gave very insightful thoughts on BioE3 and bioeconomy. And I hope all the students here are inspired by this talk, by the discussion, and will take this forward and continue to make progress. Okay, I should have put on. Then a special thanks goes to Dr. Ramchand. He not only moderated the event, but he was very instrumental in bringing all the panelists here. And also organizing this event and to make it in such a success. So thank you so much, sir. And I have to also say that he has been he has been continuously supporting CRIPS Bionest over these years, and a special thanks from me too. Yes, I would also like to thank Director of BRIC RGCB and CEO of CRIPS Bionest. Both of them couldn't be here, but their support and help has been instrumental in running the Bionest incubator and also putting together this program. And I want to give special thanks to CVJ Synthite Center and the students and CUSAT scientists who came here and participated. So, your participation is what made this a success. Yes. Yes, so everybody is included. Yeah. I mean, and this event wouldn't have been possible with the support of our staff from Bionest, organizing staff from Bionest and RGCB. So I like to thank all of you for making this event from behind the scenes a success. Okay. Participants of Ideathon, I know it was a bit small, maybe 3, but I hope that it will become better as we do more events. Thank you so much. Your participation and your creativity and inspired ideas are what make this kind of events success. Thank you. And I also want to announce, because we have not said it before, but we have a program at Bionest called co-development mode of incubation. So basically what it suggests, we envision a program where students who are not actually, who doesn't have much fund, students who can develop your product with the help of mentoring from RGCB scientists. So any of you need any kind of, so if you want to be inducted in that, please contact us. We would be very happy to do that. Sorry. So, and then finally, I would like to thank all the audience for coming here, listening to this session, and participating it to make this, because these kind of events are really meant for you and the participation. So thank you all for coming, and it has gone much beyond what we expected, but thank you for your patience and your participation. And big thanks to you for anchoring, anchoring the event. Thanks, sir. Yes.
So, but then we give double thanks for the QSAT Cochin University part and this anchoring part.
Yes, yes. And okay, that's all.
Finally, thank you for organizing everything in 2 days. Thank you.
Thank you, sir. This wouldn't have been possible without you, so thank you. And I wish you all a pleasant evening, and let's hope we will see again soon for another meeting. Thank you. Thank you.